Dear students, today we are going to discuss about the photosynthesis. Photo means sunlight, synthesis means to make or prepare a food. In the presence of sunlight, to make some amount of food material, that is photosynthesis. Generally, all animals, including human beings, is always depend upon to the plants for their food material. All green plants carry out photosynthesis. Photosynthesis takes place in green leaves in chloroplast. Green leaves in chloroplast. So photosynthesis is a physico-chemical reaction or physico-chemical process, means by which they use sunlight energy. Okay now, by which they use sunlight energy to drive the synthesis of carbohydrate or starch. To drive the synthesis of carbohydrate or starch. This is a physico-chemical reaction. Physico by means by which they use sunlight energy. Chemical to drive the synthesis of carbohydrate or starch. In this carbohydrate or starch, to have carbon, hydrogen, oxygen. Carbon, hydrogen, oxygen. Okay now, this is a photosynthesis chemical reaction. Or here 6CO2 plus 12 HCO in the presence of sunlight in the chlorophyll. C6H12O6 plus 6H2O plus 6O2. 6O2. Dear students, 6CO2 plus 12 H2O, these two are raw materials available in the atmosphere or environment. Anyhow, the CO2 or carbon dioxide to be reduced in the presence of sunlight in the chlorophyll to be make a carbohydrate or one type of starch. Then after H2O, water molecule to be released oxygen. Water molecule to be released oxygen. However, ultimately all living forms on earth depend on the sunlight for their energy. So dear students, photosynthesis is an important to two reasons. One is it is all living forms are essential all the food materials. And the second important area, it is also responsible for their plants to be release oxygen. By means transformation of physical energy is converted to chemical energy. Transformation of physical energy is converted to chemical energy. Sunlight, that is physical energy, is converted to chemical energy by using CO2 and H2O. By using CO2 and H2O. In the presence of sunlight and in the chloroplast, by using CO2 and H2O to be make a carbohydrate or starch, that is one type of glucose that having to 6 carbon of sucrose. That having to 6 carbon of sucrose. Anyhow, dear students, in their photosynthesis process to be very useful in the chlorophyll and very useful in the carbon dioxide, that is CO2. So you may have the experiment to look at in the photosynthesis process. First experiment are here to prove chlorophyll requirement. Second experiment are here to prove CO2 requirement. Okay now. So let us to prove chlorophyll requirement. Dear students, these are two leaves. These are two leaves. One is variegated leaf. Okay now. Second one is normal leaf. Right? So variegated leaf are having three types of colors. One is green in color, another one is yellow in color, another one is blue in color. By means variegated leaf containing different types of colors. And another one is a normal leaf. So normal leaf containing only one type of color, especially green in color. Why? Because dear students, variegated leaf containing chlorophyll pigments as well as another pigments that is carotenoids and xanthophylls. But normal leaf containing rich content of chlorophyll pigments. So hence normal leaf is always appeared green in color. But variegated leaf are containing low content of chlorophyll pigments and high content of carotenoids and xanthophylls. So not to be appear green in color. Okay now. Then after two leaves. So covered one black paper. Two leaves are covered black paper is one side. So take two leaves are covered one black paper. Then after these two leaves to be expressed in the sunlight. To be observed for some time. So dear students, after some time, so these two leaves presence of the one type of food material, that is starch. But normal leaf is occurs photosynthesis. Why? Because this is a green leaf containing rich content of chlorophyll pigments and to be make a own their food materials in the short period of the time. That is normal leaf is always green in color. Why? Because containing chlorophyll pigments. That is green pigments. Green color of the pigments. Chlorophyll means green color of the pigments. Normal leaf containing green colors of the pigments. That is chlorophyll. So in this chlorophyll to be easily absorbed of the CO2. So hence easily to be make a own food material. And another leaf not containing rich content of chlorophyll pigments, low content of chlorophyll pigments, not within the short period of the time to be make a own their food material, to be being of the so much time. This is a differences between variegated leaf and normal leaf. So finally, so always all green leaves easily carry out photosynthesis and within a short period of the time to be make a own their food materials. Why? Because these green leaves containing rich content of chlorophyll pigments. That is to prove chlorophyll requirement. This is a first experiment. Now let us coming to second experiment to prove CO2 requirement. In their photosynthesis process, how to utilize carbon dioxide. So this carbon dioxide finally to be make a one type of carbohydrate or one type of starch. So this is another name, that is off-leaf experiment. Okay now. So dear students, this is a normal full leaf. Then after to be cut in the same helps of the leaves. That is two off leaves. This is a one part of the off leaf. This is a another part of the off leaf. Okay now. So this is a normal green leaf. Then after to be cut two off parts. This is a one half part of the leaf. This is another half part of the leaf. Okay now. And next one to be take a one test tube. Then after one half part of the leaf to be transferred in the test tube. One half part of the leaf to be transferred in the test tube. Then after enclosed a one soft cotton, KOH soft cotton to be enclosed a one KOH soft cotton, potassium attraction soft cotton. Then after another half leaf to be expressed in the atmosphere, to be expressed in the atmosphere, to be expressed in the air, Okay now. These two are two half parts of the leaves. One half part of the leaf to be transferred in the test tube, and then after this half part of the leaf to be enclosed a some KOH soft cotton. Another half leaf is always to be expressed in atmosphere or air. Then after some time to be expressed sunlight, in the KOH test tube and normal air leaf. Okay now. So after some time, the test tube of half leaf not to be make a starch, not to be make a carbohydrate. Why? Because already this test tube is always enclosing of the KOH soft cotton. This KOH soft cotton easily absorb of the CO2. So CO2 easily absorbed of the potassium hydroxide. So hence not express of the CO2 in the half leaf, not presence of the CO2 in the half leaf. So this test tube of half leaf not to be make a own the food materials. But another leaf is to be expressed easily air. So not surrounded by KOH soft cotton. So in this time directly sunlight to be expressed of the another half leaf, and this another half leaf is always green in color and directly expressed of the sunlight. So easily to be make a own their food materials, So that is test tube half leaf not form starch. Test tube half leaf not form starch, that is negative, and another half leaf that is air form starch, that is positive. Mainly difference between are here. Test tube of half leaf enclosed the KOH soft cotton, but another half leaf not enclosed the KOH soft cotton. If KOH soft cotton is always easily absorbed of the carbon dioxide, so this carbon dioxide not expressed of the half leaf, to be stopped to the KOH soft cotton, So in this time the test tube of half leaf not form a food material, not form a starch. Only another half leaf to be make a food material, to be make a starch. So this is a to prove CO2 requirement or to prove carbon dioxide requirement. So this is introduction to photosynthesis. Okay now. Dear students, previous class we will discuss about the two experiments, that is to prove chlorophyll requirement experiment, another one to prove carbon dioxide requirement experiment. Now today we are going to discuss about the early experiments in photosynthesis. So dear students, early experiments in photosynthesis described by Joseph Priestley, John Ingenhousz, Julius von Sachs, T.W. Winkelmann and Cornelius von Ney. Dear students, first the scientist name are here Joseph Priestley. So dear students, Joseph Priestley says that prove that plants purify air by taking CO2 and releasing O2. Okay now, proved one mouse and one mint plant. One mint plant. Dear students, Joseph Priestley in the year 1770, so to be proved that the essential role of the air in the growth of the plants, and thereafter to be observed one burning candle to be a one closed space will be soon one bell jar. So after some time, this burning candle of similarly a mouse would see a suffocated with a closed space, that is a bell jar. That is bell jar. So after some time, Joseph Priestley concluded that burning candle and breathing animals, so somehow that damage of the air. But anyhow, Joseph Priestley to be placing one mint plant in the bell jar. So in this time, so the breathing animal, so always stayed alive, and the candle is always continuously burning. By means, so in those of plants to be restored in the air, However, the breathing animals and burning candles are removed. That is the bell jar experiments. Okay now, Joseph Priestley in the year between of the 1733 and 1804 to be concluded one serious experiment, that is the essential role of the air in the growth of the plants. By means, these plants to be taking of CO2 and releasing of O2. So in this phenomenon to be observed one experiment, that is a bell jar experiment. First see here. So to be taken a one burning candle in a closed space, that is to be getting of the bell jar. Then after to be taken a one animal organism, that is a mouse. So to be enclosed in a space. So in this time, the mouse suffocating in the enclosed space. So in this time, he concluded that breathing animal and burning candle, so somehow damage the air. But anyhow, so Joseph Priestley again he placed it to be one of the mint plant between of the burning candle and mouse animal organism to be a bell jar. So in this time, so the breathing animals is always alive, and the candle is always continuously burning. So finally to be concluded, plants is always stored in the air. Whatever the breathing animals and burning candles is always removed. That is Joseph Priestley experiment. And second one here, John Ingenhouse. He says that plants purify air only in the presence of sunlight. He showed that only green parts of the plants that could release O2. And the next one here, Ingenhouse used in experiment in aquatic plants. So dear students, and release of oxygen. So hence first oxygen is discovered by Joseph Priestley. Joseph Priestley in the year 1774. So Joseph Priestley already says that any green plants to be taken of CO2 and release of oxygen. But John Ingenhousz says that so the taken of CO2 and release of O2 only in the presence of sunlight, not presence of sunlight, only in the presence of sunlight. Plants purify air only in the presence of sunlight. And then after John Ingenhousz to be observed some of the aquatic plants to be applied one bright light, to be applied one bright light. So in this time small bubbles to be appeared around the small bubbles to be appeared around the green parts of the plants, green parts of the plants. These small bubbles are known as oxygen. These small bubbles are known as oxygen. Especially these bubbles are to be appeared only aquatic plants, only aquatic plants. So Ingenhousz first to be observed only some of the aquatic plants such as Pistia, Eichhornia, especially Hydrilla, especially Hydrilla. That is John Ingenhousz experiment. Okay now. Already Joseph Priestley says that any green plants to be taken of CO2 and release of O2 by means air through the CO2 and release of O2. So hence the first oxygen is discovered by Joseph Priestley in the year 1774. After some time, John Ingenhousz once again to be repeated the experiment. So all green plants the purify air only in the presence of sunlight. Then after the experiments are now to be making of some of the aquatic plants. So dear students, so John Ingenhousz to be observed some of the bubbles like structures around the green parts of the plants. These bubbles are known as oxygen. These bubbles are known as oxygen. By means green parts of the plants to be released oxygen, especially those are to be released oxygen. So in this experiment only shown in some of the aquatic plants such as especially Hydrilla. That is Hydrilla experiment. That is Hydrilla experiment. Okay now. Third one, Julius Van Sachs says that showed that green substances are located in special bodies, that is called chloroplast, where glucose is made and usually stored as starch. Stored as starch. Dear students, now third person, Julius Van Sachs. So all green plants to be prepared or make one type of glucose. This glucose is always used growing in plants. Growing in plants. So this type of food material to be available in one special bodies, that is called chloroplast. That is called chloroplast. By means some of the green color of the pigments, in the presence of sunlight to be make some amount of food materials. These food materials are developed one specialized bodies, that is called chloroplast. That is called chloroplast. That is called chloroplast. By means chloroplast are to be make one type of glucose. After some time this glucose is stored one type of starch or carbohydrate, that is C6H12O6. C6H12O6. By means finally Julius Van Sachs to be says that any green plants to be carry out photosynthesis to be make a one type of food material, that is especially starch in the presence of sunlight. In the presence of sunlight, and also to be using of green leaves. These green leaves containing some amount of chlorophyll pigment one freezer, that is one special spectrum components that illustrated by green alga, that is cladofora, and then after to be suspension aerobic bacteria. So in this aerobic bacteria to be evolved in the oxygen, only in this region of blue and red light, only in this region of blue and red light, by means the suspension aerobic bacteria to be carry out photosynthesis in the action spectrum of blue and red light regions, and first work to be action spectrum, that is TW Engelmann. He was the first person worked on action spectrum. That is credit goes to TW Engelmann. So TW Engelmann, he says that all green algae of cladofora, and similarly to be suspension of aerobic bacteria by using in the blue and red light during photosynthesis, by means during photosynthesis to be using of blue and red light. Vibsia are here 7 types of colors: violet, indigo, blue, orange, red, yellow, orange. These are the colors are here. But all green plants, especially to be using of only first taken blue light and then after to be taking red light. By means all green plants first observation of blue light and the next one observation of red light. To be observed clearly shown of the special action spectrum. And last person are here, Cornelius Varney. So he says that prove that hydrogen from a suitable oxidized compound reduces CO2 into carbohydrates, and he conducted with experiment in purple and green sulfur bacteria. Source of hydrogen in green and purple bacteria is H2S. So dear students, this is a photosynthesis chemical reaction, that is 6CO2 plus 12H2O, and to be making of C6H12O6 plus 6H2O plus 6O2. Generally, in all green plants to be carry out photosynthesis in the presence of sunlight. So dear students, in the presence of sunlight and in their chloroplast to be using two raw materials, that is carbon dioxide and water, that is CO2 and H2O. So dear students, in all green plants generally hydrogen is always donor, suitable oxidized compound, by means oxygen is always coming from the water. Prove that hydrogen from a suitable oxidized compound, So dear students, in this reaction, so the H2O, by means hydrogen is always donor to the act of donor, and in the release of oxygen, by means oxygen is always come from the water, and reduces CO2 into carbohydrate. So then after the carbon dioxide is always reduced into to make a carbohydrate. CO2 to be making a carbohydrate, H2O to be releasing of water, to be releasing of oxygen. Okay now, Cornelius Varney to be says that all green plants to be carry out photosynthesis to be requirement of CO2, H2O and chlorophyll in the presence of sunlight. So generally hydrogen is always found near to be observed some of the aerobic bacteria such as one type of purple and green sulfur bacteria. Purple and green sulfur bacteria. So in those of purple and green sulfur bacteria, so the hydrogen donor is not H2O, that is H2S, hydrogen sulfide, and oxygen to be is always sulfur, that means these organisms do not release oxygen in their photosynthesis process. In all green plants to be carry out photosynthesis to be taken of CO2 and release of O2. But few organisms, especially bacteria organisms that shown purple and green sulfur bacteria, taken up CO2 and by using not H2O, only using H2S, that is hydrogen sulfide, to be released carbohydrate, one type of carbohydrate, and also to be oxidized products, not oxidized O2, only to be oxidized some of the products, that is sulfur and sulfate, in this photosynthesis process to be released one type of sulfur and sulfates, not released oxygen, this is very very important area. All green plants in their photosynthesis process to be using H2O and releasing of oxygen, by means oxygen is always come from the water, but some of the purple and green sulfur bacteria in their photosynthesis process do not release oxygen. Why? Because not using H2O, only using one type of organic compound, that is hydrogen sulfide, H2S, hydrogen sulfide. In this time do not release oxygen, only release some of the oxidized compounds, oxidized products such as sulfur and sulfate. Okay now. So this phenomenon is created to show off all green plants and some of the purple sulfur bacteria, green sulfur bacteria to be using one type of heavy radioisotope, that is O18 heavy radioisotopes. All green plants carry out photosynthesis to be taken of CO2 and release of O2. In this phenomenon to be observed by using of one heavy radioisotope such as O18 radioisotope, and another few organisms such as purple sulfur bacteria and green sulfur bacteria, both bacteria in their photosynthesis process to be taken up CO2 and by using H2S do not release oxygen, only releasing some oxidized products such as sulfur and sulfate. So dear students, this is about the early experiments to be says that Joseph Priestley, John Ingenhousz, Julius von Sachs, T.W. Engelmann and Cornelius von Ney. Okay, now thank you. Dear students, today we are going to discuss about the structure of chloroplast. Dear students, this is a structure of chloroplast. In IP pattern, most of the time for asking in this question, that is draw a well-labeled diagram of chloroplast or explain to me the structure of chloroplast. Dear students, generally photosynthesis does takes place in green leaves of chloroplast. Photosynthesis does takes place in all green leaves of chloroplast, especially the TS of leaves containing two types of epidermis, that is upper epidermis and the second one is lower epidermis. Upper epidermis and the lower epidermis, between of the some mesophyll cells. These mesophyll cells containing large number of chloroplasts. So those mesophyll cells are here one is spongy parenchyma, second one is polysaccharide parenchyma. Polysaccharide parenchyma and spongy parenchyma to be constricted a mesophyll cells. To be constricted a mesophyll cells. Anyhow, those mesophyll cells containing larger number of chloroplasts. These chloroplasts are also occur in the green leaves. Not only green leaves, in some of the plants, parts of phylloclades and also Ophancia and Euphorbia. The stem is also containing green in color, is always to be appeared leaf-like structure. Generally, all leaves green in color. Why? Because containing chlorophyll pigment. But some of the plants of stems and also roots is also greenish in color. Why? Because containing the green color of the pigments, especially phylloclades such as Ophancia as well as Euphorbia. Okay, now, so the stem is always to be appeared green in color and platen-like structures to be carried out photosynthesis, and also photosynthetic roots such as ctenophylum and ctenospora. Generally, all roots are brown in color, but in those of plants, roots is always green in color. Why? Because containing chlorophyll pigments. Then after to be carry out photosynthesis to be make a own food material. Anyhow, dear students, photosynthesis does take place in all leaves of plants, especially all green leaves of plants. These green leaves containing a large number of chloroplasts. At the same time, some of the plant parts of stems, roots is also to be expressed leaf-like characters. Why? Because containing chlorophyll pigments. At the same time, to be carry out photosynthesis. This is very important. Okay, now, and chloroplast is a double membrane bound structure. Chloroplast is a double membrane bound structure. So, dear students, this is a chloroplast. So, these chloroplasts generally in oval in shape, round in shape, or sometimes to appear spherical in shape. Spherical in shape. The mesophyll cells are have to large number of chloroplasts. These chloroplasts are appeared to be oval, round, or spherical in shape. Spherical in shape. So this chloroplast is always a double membrane bound structure. Double membrane bound structure. So this is a outer membrane and this is a inner membrane. This is a outer membrane and this is a inner membrane, that is a double membrane bound. Chloroplast as well as mitochondria both are double membrane bound structures, and both cell organelles are largest cell organelles. Largest cell organelles. So dear students, chloroplast is a double membrane bound, that is outer membrane and inner membrane. Okay, now what is the use of outer membrane and inner membrane? Dear students, so these membranes to be trap light energy and for the synthesis of ATP and NADPH. At the same time to be provide a product. At the same time to be provide a product. That is the importance of double membrane bound. Double membrane bound. Generally, chloroplast is a double membrane bound. This double membrane bound divided into outer membrane and inner membrane. This membrane is always trapping the light energy. And for the synthesis of ATP and NADPH. ATP and NADPH. This is the importance of double membrane or outer membrane and And dear students, outer membrane and inner membrane, outer membrane and inner membrane between of the subspace, that is called periplastidial space. Periplastidial space. Outer membrane and inner membrane between of the subspace or subgap, that is called periplastidial space. Periplastidial space. And next one here, the space limited by the inner membrane of the chloroplast, that is called stroma. This is a stroma. Totally, this is a stroma. Okay, now dear students, the space limited by the inner membrane of the chloroplast. The space limited by the inner membrane of the chloroplast, that is called stroma. Okay, now the space limited by the inner membrane of the chloroplast, that is called stroma. Okay, now next one here. So the stroma, this is a stroma. The stroma containing flattened. Okay, now the stroma containing flattened membrane sacs. So these are called thylakoid. Okay, now this is a stroma, and stroma containing flattened membrane sacs. These flattened membrane sacs are called thylakoid or thylakoids. By means stroma are present in thylakoids. And next coming to thylakoids. These thylakoids are arranged in stacks like a piles of coins. Is called grana. So these are the thylakoids. These thylakoids are arranged. So these thylakoids. So especially these thylakoids are arranged in stacks like piles of coins. That is called grana. Don't confuse here. Okay, now the space limited by the inner membrane of the chloroplast, that is called stroma. Okay, now stroma containing flattened membrane sacs, that is called thylakoid. Those thylakoids are arranged in stacks like a piles of coins, that is called grana. So that is thylakoid, stroma and grana. And other important area, in addition there are flat membranous tubes called, that is stroma lamella. These are stroma lamella. So these are thylakoids. And also are in here stroma lamella. These are longer size of tubes such as, that is stroma lamella. So these are thylakoids. But this one stroma lamella, this one stroma lamella, this one stroma lamella, this one stroma lamella. In addition to the flat membranous tubules, that is called stroma lamella. This is very important area. And each thylakoid containing some of the space. This space are known as lumen. Every thylakoid containing some of the space. This space are known as lumen. Okay, now this space are known as lumen. And stroma containing only thylakoids and grana and stroma lamella, as also containing circular DNA, ribosomes and lipid particles, and as well as starch granules. starch granules. Mainly the stroma containing photosynthetic enzymes. Mainly the stroma containing photosynthetic enzymes. So hence in this time, so dark reaction takes place by stroma. Dark reaction takes place by stroma by means of non-presence of the light, but is depend on to the light. So in this time, the reduced CO2 is converted to carbohydrate. That is dark reaction. So dear students, dark reaction takes place in stroma. Stroma also containing photosynthetic enzymes. And light reaction takes place in grana or thylakoid. Why? Because so this thylakoid containing some of the space that is called lumen. This lumen is also containing photosynthetic pigments such as chlorophyll A, chlorophyll B, carotenoid and xanthophyll. Okay now. Stroma is also containing circular DNA, ribosomes and starch granules, lipid particles. At the same time, stroma containing photosynthetic enzymes. By means dark reaction takes place in stroma in this time. So to be incorporated CO2 reduced to the carbohydrate. CO2 reduced into the carbohydrate. And light reaction takes place in thylakoid or grana. Why? Because some of the thylakoids are containing some of the space. This space are known as lumen. This lumen or thylakoids is also containing some of the photosynthetic pigments. By means to be form of ATP and NADPH. ATP and NADPH is always occurs in the thylakoids or lumens of chloroplast. That is light reaction. So anyhow, light reaction takes place in thylakoid or grana of the chloroplast. Dark reaction takes place in stroma of the chloroplast. Light reaction means the directly light is given into the chloroplast. That is light reaction. So they do not directly light driven into the chloroplast. That is dark reaction. So this is a general characters in chloroplast, or this is about the structure of chloroplast. Okay, now thank you. Dear students, good morning to one and all. Previous classes we will discuss about the introduction to photosynthesis, early experiments, and what is the site of photosynthesis, that is the structure of chloroplast. Now today we are going to discuss about the photosynthetic pigments. How many pigments are involved in photosynthesis? Dear students, pigments are mainly three types. They are namely chlorophylls, carotenoids, phycobilins. Three pigments mainly are involved in photosynthesis process. They are namely first one chlorophylls, second one carotenoids, third one phycobilins. Chlorophylls are again five types such as chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. Chlorophylls are again five types such as chlorophyll A, chlorophyll B. Chlorophyll A, chlorophyll B mainly present in higher plants, and chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E present in algal members. Chlorophyll A, chlorophyll B both are present in higher plants, and again chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E are present in algal members. Chlorophyll A is the chief pigment or commonly most important pigment. Chlorophyll A is the chief pigment or common pigment in both higher plants and algal members. Both higher plants and algal members. Chlorophyll A is always expressed in bright or blue green in color. Chlorophyll B is always expressed yellow and green in color. Yellow and green in color. Chlorophyll is always to be provided one definite color in the leaves, flowers and fruits. Leaves, flowers and fruits. Especially chlorophylls are to be present in the green leaves. Chlorophylls are mainly present in the green leaves. These green leaves containing chloroplast. Chloroplast. The chloroplast of thylakoid of lumen containing photosynthetic pigments. Photosynthetic pigments such as chlorophylls, carotenoids and xanthophylls. The chlorophyll A molecular formula C55S72O5N4Mg, that is chlorophyll A containing carbon, hydrogen, oxygen, nitrogen, magnesium. So hence the color are here blue green in color. Chlorophyll B molecular formula C55S70O6N4Mg, by means this chlorophyll B is also containing carbon, hydrogen, oxygen, nitrogen, magnesium. So hence yellow green in color. So these two chlorophylls are essential in the higher plants of the leaves. And also as well as fruits. Next, coming to carotenoids. Carotenoids are of two types such as carotene and xanthophylls. Chlorophylls are of five types, but two types of chlorophylls are mainly present in higher plants, and again five types of chlorophylls are present in all algal members. But carotenoids are here two types. One is carotene, second one is xanthophylls. These carotenes and xanthophylls are present in higher plants as well as algal members. So dear students, carotene is always expressed orange and red in color. Xanthophylls is always expressed yellow and brown in color. So dear students, carotene is always expressed orange and red in color. For example, tomato, chili and carrot is always expressed in red in color. Why? Because due to the presence of one type of pigment such as carotene. Especially tomato, chilies and carrots are always appeared red in color. Why? Because due to the presence of one type of photosynthetic pigments such as carotene. And xanthophylls is always expressed yellow and brown in color. Give the example for here: banana and also mango. Banana and mango is also expressed in the yellow in color. Why? Because due to the presence of xanthophylls. Anyhow, leaves is always expressed green in color at early stage or maturity stage. Why? Because due to the presence of chlorophyll pigment. But here several flowers and several fruits are to be appeared different types of colors. Why? Because containing different types of pigments. For example, tomato, any fruit in the early stage to be appeared green in color. Any fruit to be appeared early stage in this green in color. Why? Because due to the presence of chlorophyll pigment. Especially chlorophyll A and chlorophyll B B. After maturity period, so any fruit to be appeared red in color, yellow in color, red in color, yellow in color. Why? Because due to the presence of carotene and xanthophylls, that is one type of carotenoids. That is one type of carotenoids. Okay, now clearly understood. All plants of leaves is always greenish in color. Why? Because due to the presence of chlorophyll pigment, such as chlorophyll A and chlorophyll B. But all fruits in early stage to be appeared greenish in color. Why? Because due to the presence of chlorophyll pigment. After maturity period, so some of the fruits are to be appeared yellow in color, red in color, orange in color. Why? Because due to the presence of carotenoid pigment such as carotene, either xanthophyll, and as well as flowers. Some flowers to be appeared yellow in color, some flowers to be appeared orange in color, some flowers to be appeared brown in color, sometimes some flowers to be appeared red in color, some flowers to be appeared white in color. Why? Because due to the presence of carotenoids, Okay, now next coming to phycobilins. Phycobilins are two types, such as phycoerythrin and phycocyanin. Phycobilins is one of the photosynthetic pigments. These are two types, such as phycoerythrin and phycocyanin. But these phycobilins only present in algal members, such as brown algae and blue-green algae. So these phycobilins are not present in higher plants. Don't confuse over here. Chlorophylls and carotenoids both are present in higher plants as well as algal members. Algal members is one of the plants are here. Okay, here higher plants are grown for the soil, that is terrestrial plants, but algal members is always grown in water, that is aquatic plants. Both are plants, higher plants and aquatic plants. So aquatic plants such as algal members as well as hydrilla, Pistia, Eichhornia. So but here phycobilins are present only some type of algal members such as brown algae and blue-green algae. So dear students, those two phycobilins are here, phycoerythrin and phycocyanin. So phycoerythrin and phycocyanin both are present in brown algae and blue-green algae. So depend upon to the phycoerythrin and phycocyanin. For example, brown algae. So in this brown algae, phycocyanin is less present and phycoerythrin is more present. And blue-green algae, that is like blue-green algae. So phycocyanin is more present and phycoerythrin is less present. Brown algae and red algae, especially brown algae or red algae, but especially red algae present in two types of phycobilins. But here higher amount of phycoerythrin are present in red algae, less amount of phycocyanin are present in red algae. But blue-green algae, higher amount of phycocyanin are present in blue-green algae and less amount of phycoerythrin are present in blue-green algae. So dear students, phycobilins is always depend upon to the higher amount and lower amount of phycoerythrins and phycocyanins. Okay, now next coming to pigments. These photosynthetic pigments are substances that have an ability to absorb light at different wavelengths. Dear students, pigments are one type of substances that have an ability to absorb sunlight at different wavelengths. So this different wavelengths of visible spectrum of light as well as VIBGYOR. So this different types of wavelengths of visible spectrum of light as well as VIBGYOR. So VIBGYOR means violet, indigo, blue, green, yellow, orange, red. So is highly. So dear students, so to be observed, so in those are two graphs. So first graph showing the absorption spectrum of chlorophyll A, chlorophyll B and carotenoids. Second graph showing the absorption spectrum of photosynthetic pigments and the action spectrum of rate of photosynthesis. Rate of photosynthesis. So to be observed these two graphs. First graph, this is a first graph, this is a second graph. First graph is always showing so the absorption spectrum, and second graph showing the absorption spectrum as well as action spectrum. What is the meaning of absorption spectrum? So dear students, so the absorption spectrum of graph showing the absorption spectrum of graph showing different wavelengths, different wavelengths to be chlorophyll pigments, different wavelengths by photosynthetic pigments, different wavelengths by photosynthetic pigments. So different wavelengths, that is 400 nm, 500 nm, 600 nm and 700 nm. Okay now, different wavelengths by photosynthetic pigments such as chlorophyll A, chlorophyll B, Botany-Ii. Okay now. So dear students, the first graph showing the absorption spectrum is always different wavelengths by photosynthetic pigments. By photosynthetic pigments, dear students. So this is a chlorophyll A graph. This is a chlorophyll B graph. This is a carotenoid graph. This is a carotenoid graph. So dear students, so the wavelengths of light to be described in nanometers. The wavelengths of light to be described in nanometers, such as 400 nanometers, 500 nanometers, 600 nanometers, and 700 nanometers. So, dear students, so the chlorophyll A easily absorbed by blue light. The chlorophyll A easily absorbed by blue light in 420 nanometers. Increasing of the blue light, increasing of the blue light, increasing of the rate in 420 nanometer and decreasing and again increasing of the rate. Chlorophyll A maximum absorbed by light in red light 680 nanometers and then after to be decreased. Dear students, chlorophyll A easily absorbed by blue light at 420 nanometers and again easily absorbed by red light at 680 nanometers. This is a chlorophyll A. So chlorophyll A is shown to be increasing of blue light and to be increasing of red light at 420 nanometers and 680 nanometers. Why? Because this is a graph showing 400 nanometers, 400 to 500 nanometers, 500 to 600 nanometers, 600 to 700 nanometers. So 400 to 500, that is 410, 420, 430, 440, 460, 470, 480, 490, 500. So exactly 420 nanometers will be increasing of the chlorophyll A, to be increasing of the chlorophyll A, to be increasing of the chlorophyll A, to be easily absorbed by blue light at 420 nanometers and thereafter to be decreasing or again to be increasing of the 680 nanometers to be easily absorption of red light, red light. By means, so individually the chlorophyll A is more highly absorption of blue light than a chlorophyll A, but not easily absorption of red light. So clearly understood. So chlorophyll B to be increasing other than the chlorophyll A. So this graph showing chlorophyll B increasing of the other than chlorophyll A, especially blue region, but red region is more, not more. The chlorophyll A is totally decreasing of the chlorophyll B in red region of the light. So dear students, individually chlorophyll B is more highly absorption of blue light than the chlorophyll A, but not easily absorption of red light other than the chlorophyll A. Generally color. This is a carotenoids. Carotenoids to be increasing of between of the 480 to 520 nanometers. 480 to 520 nanometers, and again decreasing of the 600 nanometers. 600 nanometers. So this is a clearly shown of absorption spectrum. The absorption spectrum of graph showing at a different wavelengths, by photosynthetic pigments such as chlorophyll A, chlorophyll B, and carotenoid. And second graph showing absorption spectrum and action spectrum. So this is a absorption spectrum, only easy absorption of by chlorophyll A. Chlorophyll A, maximum absorption by blue light and red light. The chlorophyll A, maximum absorption by blue light and red light. So in this time, to be highly increasing of the rate of photosynthesis at blue light and red light, that is 420 nanometers and 680 nanometers. Dear students, second graph showing, this is a first graph of chlorophyll A, this is a second graph of rate of photosynthesis. Rate of photosynthesis. Dear students, so the chlorophyll A to be increasing of blue region and red region, at the same time the rate of photosynthesis highly increasing of blue region and red region. By means a complete one-to-one overlapping. A complete one-to-one overlapping the absorption spectrum and the action spectrum, that is absorption of chlorophyll A and rate of photosynthesis. Rate of photosynthesis. Dear students, these graphs together also showing the rate of photosynthesis is always increasing in the blue and red regions. Blue and red regions, by means photosynthesis is always takes place in blue regions and red regions. Blue regions and red regions. Why? Because chlorophyll A is one of the most and cheap pigment for responsible light trapping or light energy. Remaining other pigments are present thallified membrane, that is chlorophyll B, carotenoids and xanthophylls. Anyhow, these other pigments is always to be protect a chlorophyll A pigment and to be trapped the light energy. And is always to be protect a chlorophyll A from photooxidation. So dear students, this is about the photosynthetic pigments, and clearly shown of two graphs. One is absorption spectrum graph, another one is action spectrum graph, and again both combined absorption spectrum graph and action spectrum graph. The absorption spectrum is the graph showing is always different wavelengths by photosynthetic pigments, and the action spectrum graph showing is always rate of photosynthesis. Rate of photosynthesis functions at different wavelengths. Rate of photosynthesis at the functions of different wavelengths, that is action spectrum. So dear students, distinguish between the absorption spectrum and action spectrum. First one, the absorption spectrum. The absorption spectrum graph showing is always different types of photosynthetic pigments is involved different wavelengths. The absorption spectrum is the graph showing at different wavelengths by different photosynthetic pigments. The action spectrum graph showing rate of photosynthesis as the function of different wavelengths. Especially the high rate of photosynthesis is including blue region and red region. Okay now. Dear students, today we are going to discuss about light reaction. So photosynthesis involves light reaction and dark reactions. Photosynthesis takes place in green leaves of chloroplast, but photosynthesis involves a light reaction and a dark reaction. what is the meaning of light reaction and what is the meaning of dark reaction? Dear students, light reaction means they directly light driven. They directly light driven. That is called light reaction. But dark reaction are here. They are not directly light driven, but depend is always light reaction products such as ATP and NADPH. That is called dark reaction. Dear students, light reaction takes place in grana or thylakoid membrane of chloroplast. Dark reaction takes place in stroma of chloroplast. So, during light reaction, light energy is absorbed by pigments found in photosystem 1 and photosystem 2. Dear students, during light reaction, light energy is easily absorbed by pigments such as chlorophylls and carotenoids. During light reaction process, light energy is easily absorbed by pigments, that is chlorophyll pigments, carotenoid pigments found in PS1 and PS2, that is photosystem 1 and photosystem 2. So in this light reactions includes light absorption, water splitting, oxygen release and the formation of high energy in ATP and NADPH. Dear students, to be observed in this light reaction or in including light absorption. First to be absorption of light and water splitting. H2O molecules is split in the light. Water molecule is split in the light, easily dissolved to be hydrogen, oxygen and also to be some amount of electrons and to be release oxygen and the formation of high energy, one type of chemical oxidation product, one type of chemical oxidation production, that is ATP and NADPH. During light reaction, the light energy is easily absorbed by some amount of pigments. These pigments is found in PS1 and PS2. So in this light reaction process, including first to be absorption of light and next one water splitting or splitting of water. In this process to be releasing of oxygen, next coming to formation of high amount of energy for the chemical oxidation products such as ATP and NADPH. ATP means adenosine triphosphate, NADPH means reduced nicotinamide adenine dinucleotide phosphate. Reduced nicotinamide adenine dinucleotide phosphate. So the pigments are organized into two distinct photochemical light harvesting complexes. The pigments such as chlorophyll pigments and carotenoid pigments are organized into two discrete photochemical. Why means these pigments are converted, light energy is converted to chemical energy, especially chlorophyll pigments, in the presence of sunlight. The chlorophyll pigments in the presence of sunlight, that is light reaction. The chlorophyll pigments in the presence of sunlight, organized into two discrete photochemicals. Why means? So the chlorophylls in the presence of sunlight, the light energy is converted to chemical energy. The light energy is converted to chemical energy by the light harvesting complex. That is LHC. So in this process, By means light energy is converted to chemical energy. So to be occur one of the complex, that is light harvesting complex. So this light harvesting complex containing within photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. This is a light harvesting complex. This diagram shown over here, light harvesting complex. Okay. Next here, the light harvesting complex are made up of hundreds of molecules of pigments, pigment molecules such as chlorophyll and carotenoid pigments, especially found in chlorophyll pigments. The light harvesting complex are made up of hundreds of pigment molecules, such as chlorophyll A, chlorophyll B, and also carotenoids bound to proteins. Chlorophyll pigments bound to proteins. So each photosystem has all the pigments. Each photosystem, this is a one type of photosystem. Light harvesting complex are known as antenna. Light harvesting complex is also known as antenna. Is containing photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. So each photosystem has all the pigments forming a light harvesting system, that is called antenna. Each photosystem has all the pigments forming a light harvesting system, that is called antenna. The light energy is converted to chemical energy within the form of light harvesting complex. This light harvesting complex is also known as antenna. This light harvesting complex or antenna to be containing photosystem 1 and photosystem 2. And photosystem 1, photosystem 2 of light harvesting complex is also containing hundreds of pigment molecules, bound to be proteins. And a special chlorophyll A forms the reaction center. This is a chlorophyll A. So this is a special chlorophyll A. Why? Because only chlorophyll A is easily absorption of the blue light and red light. So the chlorophyll A maximum absorption of blue light and red light. So in this time the chlorophyll A to be form a one center, that is known as reaction center. By means in this time the light energy is converted to chemical energy. The light energy is followed by photon. The light energy is also known as photon. The energy to be converted. Photon. The light energy is converted to be photon. The light energy is converted to be photon. Especially the light energy is easily absorption of chlorophyll A. This is a chlorophyll A reaction center. So chlorophyll A easily absorption of light energy. This light energy is converted to chemical energy. So this light energy is also known as photon. A special chlorophyll A forms the reaction center. This is a reaction center. So this reaction center is differentiated in both photosystem 1 and photosystem 2. And also photosystem 2. In photosystem 1, the reaction center chlorophyll A has an absorption peak at 700 nm, hence is called P700. Already to be observed in absorption spectrum and action spectrum to be visible spectrum. Different wavelengths of visible spectrum. So this action spectrum and absorption spectrum containing several different wavelengths of nanometers, such as 400 nanometers, 500 nanometers, 600 nanometers, and 700 nanometers. But here in this PS1, so in PS1 the reaction center, especially chlorophyll A, has an absorption peak. By means, so to be the chlorophyll A is easily absorption of higher amount of energy, higher amount of light energy. By means that is 700 nm. That is 700 nm. So dear students, in PS1 the reaction center chlorophyll A has an absorption peak at 700 nm, hence is called P700. By means the PS1 reaction center P700. Why? Because the reaction center of chlorophyll A is easily absorption of longer wavelength of red light. Longer wavelength of red light greater than or above 680 nm. Greater than or above 680 nm. So hence the PS1 reaction center is P700. P700. Next coming to in PS2 the reaction center chlorophyll A has an absorption maximum at 680 nm, hence is called P680. In PS2 the reaction center P680. Why? Because the chlorophyll A is easily absorption of shorter wavelength of red light. Shorter wavelength of red light. So less than 680 nm. Less than 680 nm. So hence the PS2 reaction center are here P680. Dear students, in PS1 the reaction center P700. Why? Because the chlorophyll A is easily absorption of longer wavelength of red light greater than 680 nm. That is P700. In PS2 the reaction center is P680. Why? Because so the chlorophyll A is easily absorption of the shorter wavelength of red light. The shorter wavelength of red light. So less than 680 nm. Less than 680 nm. So hence in PS2 the reaction center P680. So anyhow in PS1 or photosystem 1 the reaction center is P700. In PS2 or photosystem 2 the reaction center is P680. P680. Okay now. So this is a light harvesting complex diagram. So this light harvesting complex within photosystem 1 and photosystem 2. So these are pigment molecules such as chlorophyll A, chlorophyll B and carotenoids. So this chlorophyll A, mainly especially this chlorophyll A is easily absorption of light energy. This light energy is connected to photons and thereafter to be released some amount of the electrons. These electrons to be aspected by a primary substrate, that is primary electron aspector. Primary electron aspector. So by means the chlorophyll A is easily exists to be release of electron. This electron to be moving one substrate. So this substrate to be receiving of the electron. This substrate to be receiving of the electron, such as primary electron aspector. Dear students, this is about the light reaction. Okay, now thank you. Dear students, today we are going to discuss about the electron transport or photophosphorylation. Electron transport or photophosphorylation are of two types. They are namely, first one cyclic electron transport or cyclic photophosphorylation. Second one non-cyclic electron transport or non-cyclic photophosphorylation. So dear students, first we are going to discuss about cyclic electron transport or cyclic photophosphorylation. So dear students, this is a cyclic electron transport or cyclic photophosphorylation. So first one here, in cyclic photophosphorylation only PS1 is functional. In cyclic photophosphorylation only PS1 is functional. Dear students, photosystems are of two types. One is PS1, that is photosystem 1. Second one is PS2, that is photosystem 2. But here in cyclic electron transport or in cyclic photophosphorylation only PS1 is functional, not involved PS2, that is photosystem 2. In cyclic photophosphorylation only involved PS1, PS1. So dear students, the electron is circulated within the photosystem 1. The electrons is circulated within the photosystem 1. The electrons is circulated within the photosystem 1, and phosphorylation occurs due to cyclic flow of electrons. Phosphorylations occurs due to cyclic flow of electrons between cytochrome B6 and cytochrome F. The electron is circulated within the photosystem 1, and phosphorylation occurs due to cyclic flow of electrons. Cyclic flow of electrons between cytochrome B6 and cytochrome F. Phosphorylation means, so in this process, the synthesis of ATP molecules from ADP and inorganic phosphate in the presence of light. This is the light reaction. In the presence of light, that is called phosphorylation. That is called phosphorylation. That is photophosphorylation. Phosphorylation means, in this process, forming a ATP by the cells, that is chloroplast or mitochondria, that is called phosphorylation. That is called phosphorylation. But photophosphorylation, in this process, the synthesis of ATP molecules from ADP plus inorganic phosphate in the presence of sunlight. In the presence of sunlight, that is called photophosphorylation. Phosphorylation means, in this process, the forming of ATP by those of cells, that is chloroplast and mitochondria, that is called phosphorylation. But photophosphorylation, in this process, the synthesis of ATP molecules from ADP, adenosine diphosphate, and IP, inorganic phosphate, in the presence of sunlight, that is photo, in the presence of sunlight, that is called photophosphorylation. Okay now, the electron is circulated within the photosystem 1, and phosphorylation occurs due to cyclic flow of electrons. And next coming to, in this process, the electrons from the reaction center of PS1. So in this process, the electrons from the reaction center of PS1. So the reaction center are here P700. Chlorophyll A, in a special chlorophyll A to be formed from reaction center. So this reaction center of chlorophyll A is easily absorption of longer wavelength of red light. That is 700 nm. That is 700 nm. Above the 680 nm, above the 680 nm or greater than 680 nm. So in this time, the reaction center are named as P700. Okay now, in a special chlorophyll pigment, that is chlorophyll A, is easily absorbed from the longer wavelength of red light at 700 nm. By means above the 680 nm or greater than 680 nm. So in this time, so the reaction center, in this time the PS1 reaction center is P700. So in this process, the electrons from the reaction center of PS1, electrons to primary electron aspirator. In this process, the electrons move from PS1 and then after to be reached to primary electron aspirator. That is peridoxine. So next earlier now, the excited electrons is cycled back to PS1. So in this excited electron again came back to reach out the reaction center P700 or PS1 through the electron transport system. The excited electrons is cycled back. Back to PS1 through the electron transport system. Anyhow, in this process, only formation of ATP molecule. process only formation of ATP molecule between of the cytochrome B6 and the cytochrome F, and not formation of NADPH. There is no not formation of reduced NADP. Reduced NADP. So, dear students, this is a cyclic electron transport or cyclic photophosphorylation. So, to be explained diagrammatic representation. So, dear students, so 1A special chlorophyll A to be absorbed to longer wavelength of red light at 700 nm, that is above the 680 nm or greater than 680 nm. So, in this time the P700 to be act of reaction center. So, this reaction center of P700 to be release of electrons. So, these electrons to be receiving of the P700. So, in this time to be excited in this time. So, to be excited and then move to electrons first receiving of primary electron aspector. Primary electron aspector. By means in this PS1 system, the chlorophyll A easily absorbs the longer wavelength of red light at 700 nm. At 700 nm. By means causing the electrons excited, and then after move to electrons in the primary aspector. Then after move to electrons from the primary aspector, that is pyridoxine. So P700. So to be excited, those electrons to become excited and jump into the primary aspector. So the primary aspector is pyridoxine. By means primary aspect of pyridoxine receiving of the electron. Okay, now the PS1 of P700 to be excited and then after jump to the electrons. So these electrons will be receiving of primary aspector, that is pyridoxine. So pyridoxine is the primary electron aspector in the cyclic electron transport system or cyclic photophosphorylation. Cyclic electron transport or cyclic photophosphorylation, the primary electron aspector is pyridoxine. So next coming to the electron move to cytochrome complex, that is cytochrome B6 and cytochrome F. So between of the plastophenone. So then after the electron transferred into cytochrome complex, first transfer to cytochrome B6. So in the cytochrome B6 again get excited and to be formation of ATP, and thereafter to be releasing of again electron. This electron to be reached plastophenone, then after to be electron reach of the cytochrome F. So finally this electron to be reach plastocyanin. So this plastocyanin again to be release of electron. This electron came back to again to be reach of photosystem 1 of P700. This is a one cyclic flow. By means electrons. So first of all electrons to be released P700, again to be reach P700. First the electrons to be released the reaction center of P700, and then after finally to be again reach came back to the reaction center of P700. So dear students, in this cyclic electron transport or cyclic photophosphorylation to be formation of ATP molecules between of the cytochrome B6 and cytochrome F, there is no formation of NADH. Anyhow, in this cyclic electron transport system or cyclic photophosphorylation, only involved PS1 or photosystem 1, only involved one reaction center, that is P700. So in this cyclic photophosphorylation, the primary electron aspector is pyridoxine. in this cyclic photophosphorylation only formation of ATP molecules between of the cytochrome B6 and cytochrome F. By means in this cyclic electron transport or cyclic photophosphorylations, electrons move from one carrier to another carrier, especially it is concerned to cytochrome complex. It is concerned to cytochrome complex. So anyhow, so the electrons easily released to P700, again reach to P700. This is a one cyclic flow. So between of the one electron receiver, that is plastocynin. So anyhow, in this cyclic photophosphorylation, to be include photosystem 1 or P700 and pyridoxine, cytochrome B6, plastoquinone, cytochrome F and plastocynin. This is a plastoquinone, this is a plastocynin, this is a cytochrome B6, this is a cytochrome F. So in this cyclic photophosphorylation, the primary electron aspect is pyridoxine. Okay, now this is about the cyclic electron transport or cyclic photophosphorylation. Okay, now dear students, previous class we will discuss about cyclic electron transport or cyclic photophosphorylation. Now today we are going to discuss about the non-cyclic electron transport or non-cyclic photophosphorylation. Non-cyclic photophosphorylation are also known as Z scheme. Cyclic electron transport or cyclic photophosphorylation are involved only one photosystem, that is PS1, photosystem 1. But in non-cyclic photophosphorylation are involved two photosystems. One is photosystem 2, second one is photosystem 1. But dear students, first to be operating PS2, then after to be operating PS1. In cyclic photophosphorylation or cyclic electron transport are involved only one type of photosystem, that is photosystem 1. But in non-cyclic electron transport or non-cyclic photophosphorylation are involved two photosystems, that is photosystem 1 and photosystem 2. But first the function in PS2 and the next function in PS1. Okay, now next coming to the PS2 reaction center P618, the PS1 reaction center P700. PS2 reaction center P680, PS1 reaction center P700. PS2 primary electron aspect is PO510. PS2 primary electron aspect is PO510. This is a PO510. PS1 primary electron aspect is pyridoxine, either A0. PS1 primary electron aspect is A0 or pyridoxine, that is FD. Pyridoxine. Okay, now coming to discuss about overall non-cyclic electron transport or non-cyclic photophosphorylation. So dear students, so this is a photosystem 2, this is a photosystem 1. But here first functioning in photosystem 2, next functioning in photosystem 1. Two photosystems are functioning in non-cyclic electron transport or non-cyclic photosphorylation. Dear students, A special chlorophyll A to be formed one reaction center, that is P680. P680. A special chlorophyll A to be formed of one reaction center, that is P680 in the photosystem 2. By means the chlorophyll A easily absorption of short wavelength at 680 nanometers, less than 680, between of the 650 to 680, between of the 650 to 680. So this 680 is the reaction center in PS2. Okay, now the chlorophyll A absorption of sunlight at the shorter wavelength of light, at a visible spectrum in 680 nanometers. So hence the PS2 reaction center is P680. P680. So in this time P680 get excited, and then after liberated to electrons. So these electrons to be accepted one carrier, that is PO15. By means electrons coming from PS2 of P680. So these electrons to be accepted by or to be receiving by one iron rich carrier protein, that is PO15. Okay, now so in this PO15 to be receiving of 2 electrons. Dear students, in the non-cyclic photophosphorylation, especially PS2, for every time to be release of 2 electrons. So those 2 electrons to be receiving PO15. So in this PO15, this is a primary electron hospital. In PS2, first primary electron hospital is PO15, PHEO, that is PO15. PO15 to be receiving of electrons from the P680. By means electrons moves from P680 to be accepted by PO15. So PO15 is the primary electron aspector. In PS2. So in those of 2 electrons again to be enter into the phenol cycle, that is QA and QB, that is phenol cycle. So in this phenol cycle to be receiving of the 2 electrons. So these 2 electrons to be enter into the plastophenol. So in this time the plastophenol to be drawing up 1 electron and to be receiving of 2 protons in the stroma, from the stroma. So in this time plastophenol is converted to plastophenol, that is PQH2. This PQH2 is again reoxidized into plastophenol or plastosemi-phenol. So in this time again the plastosemi-phenol to be receiving of the cytochrome B6F complex. Cytochrome B6F complex. At the same time, to be releasing of two protons for the lumen of thylakoid. For the lumen of thylakoid. So in these protons are synthesized by ATP molecules. In these protons are synthesized by ATP molecules. Okay now. So dear students, plastopinone is also one of the iron rich electron protein. At the same time, cytochrome B6, cytochrome F, both are also iron sulfur proteins. Iron sulfur proteins. Next, those electrons to be enter into the plastocyanin. Plastocyanin. This is a sulfur protein. This is a sulfur protein or copper protein. This is a sulfur protein or copper protein. So this to be act of mobile electron carrier. Mobile electron carrier. Why? Because in those are two electrons to be entered of the PS1 of P700. P700. PS1 of P700. So in the PS2 and PS1 to be electrons is always transported within the one electron carrier, that is called plastocyanin. So hence the namely mobile carrier, mobile electron carrier, mobile electron carrier. Plastocyanin is a mobile electron carrier between PS2 and PS1. PS2 and PS1. That is plastocyanin. Now next coming to PS1 of P700. So again the chlorophyll A is easily observed of longer wavelength of red light in visible spectrum at 700 nm, that is above the or greater than 680 nm. So in this time, so hence the PS1 reaction center P700. So this P700 again get excited and liberated to be electrons. So these electrons to be reached to be accelerated first to A0, then after electrons to be reached A1, then after electron to be reached Fx, and then after electrons to be reached Fa, Fb complex. Finally electrons to be reached pyridoxine. Pyridoxine, this is a primary electron transporter in the PS1. But these A0, A1, Fx, Fa, Fb, these are internally electron carriers. But external electron carrier are here Fd, that is pyridoxine. That is pyridoxine. So in this pyridoxine to be involved, electrons to be moved from the plastopinone and again to be forming of one ATP molecules between of the cytochrome B6 and cytochrome F. Cytochrome B6 and cytochrome F in their cyclic photophosphorylation. So in this pyridoxine to be receiving of electrons, in those of electrons to be entered into the FNR. Flavinonucleotide, reduced flavinonucleotide. FNR means reduced flavinonucleotide. So in those of reduced flavinonucleotide containing one type of coenzyme, that is NADP+. This NADP+ will be receiving of electrons. Then after to be reduced to NADPH+. So every time to be receiving of the electrons. So in this time to be losing of electrons, the PS1. To be losing of the electrons in PS1. Why? Because NADP, oxidized NADP get reduced to NADPH plus H+. NADPH plus. At the same time to be synthesized ATP molecules. So dear students, in non-cyclic photophosphorylation to be forming ATP molecules, and NADPH molecules. To be forming ATP molecules and NADPH molecules. So to be clearly understood here, this is a non-cyclic photophosphorylation involved PS2, PS1. But cyclic photophosphorylation, the PS1 only involved to the formation of ATP molecules, not formation of NADPH molecules. But here, clearly, don't confuse here. This is is a non-cyclic photophosphorylation. This one PS2, this one PS1. But clearly over here ATP molecules are formed PS2 and PS1, and the NADPH molecules to be formed non-cyclic photophosphorylation of PS1. End of the reaction. End of the non-cyclic photophosphorylation. End of the non-cyclic electron transport or non-cyclic photophosphorylation to be forming of NADPH molecules. NADPH molecules. Okay now. But cyclic electron transport and cyclic photophosphorylation, only formation of ATP molecules. Only formation of ATP molecules same between of cytochrome B6 and cytochrome F, but not formation of NADPH molecule. But in non-cyclic photophosphorylation, the end of PS1 by means involved PS2 and PS1, but end of the PS1 to be formation of NADPH. To be formation of NADPH by means the PS1 to be losing of electrons. So in this time the PS2 containing water molecules, these water molecules occurs in the lumens of thylakoids. So in this time the water molecule is split with the help of manganese, calcium and chlorine. So we will be forming of 2 protons, 2 electrons and oxygen. So dear students, splitting of water or oxygen leveling complex is closely associated within the PS2. Splitting of water or water splitting and oxygen leveling complex are closely associated within the PS2. The splitting of water is present in lumina of thylakoids. For example, one water molecule is split with the help of manganese Mn2+, calcium Ca+2, chloride Cl-2 to split to release 2 protons, 2 electrons and of oxygen, molecular oxygen. This is the end product. This is a end product of photosynthesis. Oxygen is to be end product of photosynthesis. So these 2 protons occurs in the lumina of thylakoids, but these electrons are again to be receiving of PS2 P68B. By means, so those electrons to be replaced by PS2 and removed by PS1, but these electrons is always provided by PS2. If 2 water molecules, so 2 water molecules is split within the help of manganese, calcium, chlorine to be release of 4 protons, 4 electrons and 1 oxygen molecule. This process is known as splitting of water or photolysis. Photolysis means the presence of sunlight. 1 water molecules or 2 water molecules is split in the presence of sunlight. That is photolysis. Or splitting of water to be release protons, electrons and oxygen. So this splitting of water is also responsible for oxygen leveling complex. This splitting of water is also responsible for oxygen leveling complex, short form OEC. By means it convinced in involving of oxygen. This oxygen to be end product of the photosynthesis. Okay, now this is a non-cyclic electron transport or non-cyclic photophosphorylation. So, dear students, what is the differences between cyclic electron transport or non-cyclic electron transport? So, first one, cyclic electron transport. So, cyclic electron transport or cyclic photophosphorylation are involved only Next, cyclic photophosphorylation. The PS1 primary electron aspect is A0 or pyridoxine. In non-cyclic photophosphorylation, PS2 primary electron aspect is PO15. So cyclic electron transport are formed only ATP molecules. Non-cyclic photophosphorylation are formed ATP molecules and NADPH molecules. So these are some differences between cyclic photophosphorylation and non-cyclic photophosphorylation. Another important are here, cyclic photophosphorylation does not splitting of water, does not evolving of oxygen, but non-cyclic photophosphorylation to be present splitting of water by means evolving of oxygen, to be end product of oxygen to be photosynthesis, this oxygen to be released non-cyclic electron transport. And cyclic electron transports, electrons is always moves closely circuits. Electrons is always move from closed circuit. Electrons is always move from closed circuit. Electrons is always move from closed circuit. But non-cyclic electron transport or non-cyclic photophosphorylation, electrons move from zigzag manner. Electrons move from zigzag manner and it will be appeared Z shape. That is called Z scheme. Non-cyclic electron transport from zigzag manner is called Z scheme. Cyclic electron transport, the electrons moves from closed circuit. By means, some of the electrons to be released to P700, the reaction center of P700, again to be reached the reaction center of P700, that is a closed circuit. But non-cyclic electron transport, the electrons move from zigzag manner. The electrons move from zigzag manner or to be appeared Z shape or N shape. So this non-cyclic electron transport or this non-cyclic photophosphorylation are also known as Z scheme. And non-cyclic photophosphorylation includes splitting of water, release of oxygen, and formation of ATP molecules and formation of NADPH molecules. So these are some differences between cyclic electron transport or non-cyclic photophosphorylation, non-cyclic electron transport. And some of the differences between photosystem 1 and photosystem 2. So first one here, photosystem 1. Photosystem 1 only involved cyclic photophosphorylation, but photosystem 1, photosystem 2 both are involved non-cyclic photophosphorylation. Okay, now photosystem 1 only located in stroma lamella or grana lamella, but PS2 are located only stroma thallicae. PS1 are located in stroma thallicae or grana thallicae, but PS2 are located only stroma thallicae. PS1 containing 100 molecules of chlorophyll A, chlorophyll B. PS2 containing 250 molecules of chlorophyll A and chlorophyll B. And PS1 containing rich content of carotenes, PS2 containing rich content of xanthophylls. So in this PS1, the chlorophyll A chlorophyll B ratio 4:1. In PS2, the chlorophyll A chlorophyll B ratio 1:1. PS1 only absorbs a longer wavelength of red light at 700 nm. PS2 only absorbs a shorter wavelength of light at 680 nm. These are some differences between PS1 PS2. Anyhow PS1 primary electron aspect is A0 or pyridoxine, but PS2 primary electron aspect is PO15. PO15 is one special type of chlorophyll A, but chlorophyll A containing center of the magnesium atoms and surrounded by 4 nitrogen molecules. But here PO14 of chlorophyll A containing center of the magnesium atom but surrounded by 2 hydrogens. This is the difference between chlorophyll A and PO14. And plastocyanin are involved in both PS1 and PS2, that is cyclic protophosphorylation and non-cyclic protophosphorylation. Plastocyanin to be act of mobile electron carrier. Overall, in cyclic protophosphorylation and non-cyclic protophosphorylation are involved some electron carriers such as quinone, plastoquinone, and cytochrome complex, that is cytochrome B6 and cytochrome F, and plastocyanin. But PO14 and pyridoxine both are primary electron aspectors. Anyhow, the PS1 reaction center is P700, the PS2 reaction center is P680. So this is about the non-cyclic electron transport or non-cyclic protophosphorylation or jet stream. Okay now? Dear students, today we are going to discuss about the chemi-osmotic hypothesis. Already previous classes we will detailedly discuss about light reaction in those of non-cyclic electron transport or non-cyclic protophosphorylation and cyclic electron transport or cyclic photophosphorylation. So these two phosphorylations to be released some amount of protons. So these protons to be utilized ATP. These protons to be utilized ATP production. So dear students, chemi-osmotic hypothesis is proposed by Peter Mitchell in the year 1961. This hypothesis or this theory clearly explains proton concentration gradient in thylakoid membranes as well as lumen. So dear students, this is one of the thylakoid. Generally, one specific chloroplast containing one fluid matrix, that is called stroma. So this stroma containing flattened shaft-like structures, that is known as thylakoids. Those thylakoids are arranged on piles of stacks like coins, that is called grana. But here this is a thylakoid. So thylakoids are present in stroma of chloroplast. So dear students, this is a outer membrane thylakoid. This is a inner membrane thylakoid. So this thylakoid containing some empty space or cavity, that is called lumen, and this is a stroma, that is one fluid matrix, that is called stroma. And also, so this lumen containing OEC, oxygen evolving complex, or photolysis of water by means inner membrane of thylakoid are containing oxygen evolving complex or photolysis of water, and outer membrane of thylakoid containing NADP reductase enzyme. So dear students, already previous classes we will detailedly discuss about non-cyclic electron transport or cyclic electron transport. But here first we will discuss about non-cyclic electron transport. So dear students, so non-cyclic electron transport. So in this transport to be present two photosystems. One is photosystem 2, another one is photosystem 1. First will be present photosystem 2, second will be present photosystem 1. So photosystem 2, so to be observed shorter wavelength of red light, approximately 680 nm, to be excited and jump to be released some amount of the electrons. These electrons to move one carrier to another carrier, finally to be reach photosystem 1. So dear students, PS2 electrons to be passing through electron carriers, finally to be reach of PS1. So this is a one type of electron transport chain. So this electron transport chain mainly involved quinone, plastoquinone, cytochrome B6F complex and plastocyanin. But here mainly cytochrome B6 and F complex. So this cytochrome B6 and F complex is always transport of electrons and translocation of protons from stroma to lumen. This is the important function are here, cytochrome B6 and F. Cytochrome is one of the iron sulfur protein. This iron sulfur protein of cytochrome B6 is always to be helping transport of electrons one carrier to another carrier, and then translocation of protons from stroma to lumen. This is a stroma. Thylakoid membrane surrounded by stroma. This is a lumen. So protons to be move stroma to lumen. How many protons? Each time 2 protons. One pair of protons. So dear students, one pair of protons to be moved stroma to lumen with the help of cytochrome B6 F complex. At the same time, the PS1 is also to be carrying electrons one carrier to another carrier between of the ferridoxine and plastoquinone, cytochrome B6, cytochrome F and plastocyanin. At the same, the cytochrome B6 and the cytochrome F are involved within the help of electron transport and proton translocations. So these protons safely move to stroma to lumen. But here first observed non-cyclic electron transport of PS2 and PS1. Between of PS2 and PS1, the mainly cytochrome B6 to be carrying of 2 protons from stroma to lumen. So the lumen containing 2 protons. This is a first step. And second step over here, let us. So already one water molecule splitted in this place, that is expressly inner membrane of thylakoid. The inner membrane of thylakoid to be containing one water molecule near the PS2 complex. By means PS2 is closely associated with the one water molecule is responsible for oxygen leveling complex, short form OEC. Okay now. So this one H2O molecule to be splitted in the presence of light, to be resulted 2 electrons, 2 protons of O2 molecule, that is called photolysis of water. So this H2O molecule to be produced how many protons? 2 protons, that is one pair of protons. So these protons to be occupied in the lumen. But these 2 electrons, so to be receiving of the PS2 and half OT to be released in the stroma to be utilized photosynthesis process. This is the end of the product. So O2 is one of the end product for photosynthesis. This O2 to be released in the stroma, but protons are occupied in the lumen. these electron, one pair of electrons to be receiving of PS2, and this PS2 to be receiving of electrons and it will be transported through electron chain system within the PS1, within the PS1. So already in this lumen containing two protons, at the same time the water molecule to be produced two protons. So totally 4 protons are being of the lumen, So in this time the lumen containing higher proton concentration. So the higher proton concentration containing lumen. So in this time within the help of one factor, one ATP synthase enzyme, within the help of one ATP synthase enzyme or one factor, that is coupling factor. So coupling factor is located between of the outer membrane, inner membrane, and also stroma. Okay now. So this ATP synthase enzyme or coupling factor containing two parts. One is F0, second one is F1. F0 that is stop, F1 that is head. This is a head part, this is a stalk part or tail part. Already lumen containing several type of protons, approximately 4 protons are here. In this time the lumen containing higher concentration gradient, higher proton concentration gradient. The higher proton concentration protons to be moved inside of the stroma within the help of ATP synthase enzyme or coupling factor. The ATP synthase enzyme or coupling factor consists of two parts. Okay now. One is F0, second one is F1. F0 are here stalk or tail, F1 are here head. Okay now. So these protons to move down concentration gradient. These protons to move down concentration gradient from lumen to stroma. In this time to be released some amount of the energy. So this energy are replaced adenosine diphosphate and inorganic phosphate to be make one ATP molecule with the help of ATP synthase enzyme. By means protons to be moved higher concentration region to lower concentration region. So here where higher concentration region that is lumen, lower concentration region that is stroma. Within the help of ATP synthase enzyme or coupling factor. Okay, clearly understood. Lumen containing higher concentration, stroma containing lower concentration. Why? Because lumen containing higher concentration already to be occupied protons. So these protons are having to higher concentration gradient. So the higher concentration gradient of protons to be moved downwards of the lower concentration gradient of stroma with the help of ATP synthase enzyme. This ATP synthase enzyme consists of two parts, F0, F1, that is stop and that is head. So F0 is always embedded in the membrane. With the help of facilitated membrane. Or facilitated diffusion within the proton membrane. And F1 is always to be at the catalytic site. By means is always synthesize ATP molecules in this place. So hence, so the higher concentration of protons to be moved downwards the lower concentration of the region, that is protons to be moved lumen to stroma with the help of ATP synthase enzyme or coupling factor. So anyhow, in non-cyclic electron transport or non-cyclic photophosphorylation, to be released 4 protons, and cyclic electron transport or cyclic photophosphorylation to be released 2 protons. Totally lumen containing how many protons? 6 protons. But clearly shown are here 4 protons or H plus ions. Clearly shown are for non-cyclic electron transport. And 3 protons to be make a 1 ATP molecules. So here 6 protons to be make how many ATP molecules? That is 2 ATP molecules. Every time 3 protons are to be make 1 ATP molecules. 1 ATP molecules to be synthesized with the help of 3 protons. So this hypothesis or this theory is clearly proposed by Peter Mitchell in this year 1961. So dear students, so this theory is clearly help to understand how is synthesis of ATP molecules in non-cyclic electron transport or cyclic electron transport. Anyhow, protons to be make ATP molecules in the stroma. Why? Because stroma containing thylakoids as well as containing photosynthetic enzymes. And next coming to our here, so the inner membrane thylakoid containing OEC or photolysis of water, but outer membrane thylakoid one type of enzyme, that is NADP reductase enzyme. So this NADP reductase enzyme to be containing first of all NADP. This NADP again to be receiving of protons, then will be converted to NADPH plus H plus. Why? Because so these protons to be released some amount of the energy. This energy to be utilized adenosine diphosphate and inorganic phosphate to be make adenosine triphosphate. Then after these protons to be joining NADP to be converted NADPH plus H plus with the help of NADP reductase enzyme. So dear students, overall are here. This is a outer membrane thylakoid, this is a inner membrane thylakoid, and thylakoid containing some empty space or cavity, that is called lumen. So NADP reductase enzyme are located outer membrane thylakoid. OEC or photolysis of water are located inner membrane of thylakoid. The lumen containing some amount of the protons. The stroma containing ATP synthase enzyme. So ATP to be made in the stroma. Okay now. Dear students, previous classes we will discuss about light reaction. Includes non-cyclic electron transport or non-cyclic photophosphorylation and cyclic electron transport or cyclic photophosphorylation. Dear students, the end products of light reaction is ATP and NADPH. ATP and NADPH. ATP and NADPH are also known as assimilatory power products. So the light reaction is completed. Today we will discuss about dark reaction. Light reaction is always depend on the light, but dark reaction is not depend on sunlight, but depend on the light reaction end products such as ADP and NADPH. Light reaction end products are here ATP and NADPH. This ATP and NADPH are using in this dark reaction process. The CO2 is reduced into carbohydrate. The CO2 is reduced into carbohydrate by using ATP and NADPH. ATP and NADPH. Okay, clearly understood. Light reaction is always depend on the light. Already in previous classes we will detailedly discuss about light reaction of non-cyclic photophosphorylation and cyclic photophosphorylation. At the same time, chemi-osmotic hypothesis. Dark reaction over here is not depend on light, indirectly depend on sunlight, by means to be depend on light reaction end products such as ATP and NADPH. ATP and NADPH. So these ATP and NADPH are also known as assimilatory power products. Assimilatory power products. In this dark reaction process, the CO2 is reduced into carbohydrate by using light reaction end products, that is ATP molecules and NADPH molecules. ATP molecules and NADPH molecules. Light reaction mainly takes place in grana of thylakoids, but dark reaction takes place in stroma. Dark reaction takes place in stroma. Why? Because the stroma containing photosynthetic enzymes. The stroma containing photosynthetic enzymes. Don't confuse over here. Light reaction and dark reaction both are occurs in the granules of chloroplast. But light reaction takes place in grana or thylakoid membranes, and dark reaction takes place in stroma. Okay now. So in the dark reaction, to be involved C3 plants, C4 plants, as well as CAM plants. CAM plants, by means in their dark reaction process, some of the plants to be utilized 3 carbon compounds, to be utilized 4 carbon compounds, to be utilized again 4 carbon compounds, finally to be making some amount of food material such as one type of carbohydrate or one type of exose sugar. Exose sugar. Now we are going to discuss about C3 cycle. So dear students, C3 means some plants to be form of, first to be form of 3 carbon compound. 3 carbon compound. First to be form of 3 carbon compound. Some plants first to be form of 3 carbon compound. So hence in these plants are named as C3 plants. In this cycle are named as C3 cycle. This C3 cycle first will be observed by Melvin-Calvin. So hence this cycle are named as Melvin-Calvin cycle. So dear students, Melvin-Calvin first to be observed one green algae such as chlorella, to be using C14 radioactive isotope. After some time to be making it some amount of food material in the presence of assimilatory power. In the presence of assimilatory power, in the presence of explanatory power. So dear students, C3 cycle are also named as Melvin-Calvin cycle or PCR cycle, that is photosynthetic carbon reduction. Photosynthetic carbon reduction. Photosynthetic carbon reduction cycle, PCR. C3 cycle are named as Melvin-Calvin cycle or PCR cycle. C3 cycle means some plants first to be form of 3 carbon compound of organic acid. Such plants are named as C3 plants. Such 3 carbon compounds of organic acid are named as C3 cycle. In this C3 cycle, first to be observed Melvin-Calvin. So hence the namely Melvin-Calvin cycle. Okay, now Melvin-Calvin cycle. C3 cycle or Melvin-Calvin cycle or PCR cycle or PCR cycle. PCR cycle. PCR means photosynthetic carbon reductive. Photosynthetic carbon reductive cycle. Photosynthetic carbon reductive cycle. Photosynthetic carbon reductive cycle. So that is PCR cycle. C3 cycle or Melvin-Calvin cycle or PCR cycle, that is photosynthetic carbon reductive cycle, are shown in C3 plants. Are shown in C3 plants. C3 cycle or Melvin-Calvin cycle are having 3 steps are here. First step are here carboxylation. Second step are here reduction. Third step are here regeneration. Regeneration. C3 cycle or Melvin-Calvin cycle or PCR cycle or involves 3 steps or 3 pages are here. First stage or first step or first phase are here carboxylation. Second step, second stage or second phase are here reduction. Third stage or third phase or third step are here regeneration. So dear students, first we are going to discuss about first 2 steps. First one carboxylation, second one reduction. Second one reduction. So dear students, this is a C3 cycle or Melvin-Calvin cycle. This is a C3 cycle or Melvin-Calvin cycle. Dear students, already the chloroplast of stroma containing 6 molecules of ribulose 1,5 bisphosphate. C3 cycle or Melvin-Calvin cycle occurs in the chloroplast of stroma. So this stroma already would be containing 6 molecules of ribulose 1,5 bisphosphate, that is RUBP. That is RUBP. So this RUBP to be aspected by 6 molecules of CO2 from atmosphere. 6 molecules of CO2 from atmosphere. So 6 molecules of CO2 are available in the atmosphere. So this 6 molecules of CO2 first aspected by 6 molecules of ribulose 1,5 bisphosphate, that is 6 molecules of RUBP. This 6 molecules of RUBP containing 5 carbon compounds. Compounds are here 5 carbon compounds, but molecules are here 6 molecules. So molecules are here 6 molecules, carbon compounds are here only 1. Okay, now 6 molecules of RUBP to be receiving of 6 molecules of CO2. So with the help of one enzyme, that is Rubisco. So here primary CO2 aspect is RUBP. Primary CO2 aspect is RUBP. By means 6 molecules of ribose 1,5 bisphosphate already would be containing chloroplast of stroma. In this time 6 molecules of CO2 available for atmosphere. So this atmosphere of CO2 first aspected by 6 molecules of RUBP with the help of enzyme Rubisco. With the help of enzyme Rubisco, that is RUBP carboxylase oxidase. Rubisco means RUBP carboxylase oxidase. In this C3 cycle only utilized RUBP carboxylase. Then will be converted 12 molecules of 3-phosphoglyceric acid. 12 molecules of 3-phosphoglyceric acid. Why? Because 6 molecules of RUBP is combined with 6 molecules of CO2. Then after within the form of 12 molecules, 6 molecules plus 6 molecules, that is 12 molecules of 3-phosphoglyceric acid. This 3-phosphoglyceric acid containing 3 carbon compounds. This is a first stable compound. In C3 cycle, the first stable compound, that is 3-PGA. 3-PGA means 3-phosphoglycerate or 3-phosphoglyceric acid. So this reaction are named as carboxylation. This is a first step over here. Once again recall, 6 molecules of RUBP will be containing the stroma of chloroplast. 6 molecules of CO2 available from atmosphere. This 6 molecules of CO2 first aspected by 6 molecules of RUBP with the help of enzyme Rubisco to be formed 12 molecules of 3-phosphoglycerate or 12 molecules of 3-phosphoglyceric acid. In this reaction are named as carboxylation. 6 molecules of RUBP and 6 molecules of CO2 will be combined with the form of 12 molecules of 3-phosphoglyceric acid. So 3-phosphoglyceric acid or 3-phosphoglycerate are containing 3 carbon compounds. So hence in this cycle are named as C3 cycle. This reaction are named as carboxylation. This is a first step. So here in C3 cycle, primary CO2 aspect is RUBP. So in C3 cycle, first stable compound is 3-PGA, 3-phosphoglycerate. By means 1 CO2 is required. 3 ATP molecules, 2 NADPH molecules. 3 ATP molecules and 2 NADPH molecules. Next we are going to second step over here, reduction. So dear students, 12 molecules of 3-phosphoglyceric acid or 3-phosphoglycerate with the using of light reaction end products. First using ATP molecules, then after to be using NADPH molecules. Okay now. So by means reduction reaction completed 2 steps. Reduction reactions are to be completed 2 steps, but carboxylation reaction will be completed only 1 step. But reduction reactions will be completed in 2 steps. First step over here, 12 molecules of 3-phosphoglyceric acid or 12 molecules of 3-phosphoglycerate is reacted with 12 molecules of ATP molecules, 12 ATP molecules to be formed 12 molecules of 1,3-bisphosphoglyceric acid. 12 molecules of 1,3-bisphosphoglyceric acid. So this reaction is catalyzed by the enzyme phosphoglycerokinase. This is a first reduction reaction. by means 12 molecules of 3-phosphoglycerate are to be using light reaction end product such as 12 molecules of ATP, 12 molecules of ATP. So hence in this time 12 molecules of 3-phosphoglycerate to be formed 12 molecules of 1,3-bisphosphoglyceric acid, 12 molecules of 1,3-bisphosphoglyceric acid. So in this reaction is catalyzed by the enzyme such as phosphoglycerokinase, phosphoglycerokinase. So next coming to second reduction reaction, 12 molecules of 1,3-bisphosphoglyceric acid is converted to 12 molecules of glyceraldehyde 3-phosphate. 12 molecules of glyceraldehyde 3-phosphate. So dear students, 12 molecules of 1,3-bisphosphoglyceric acid are using light reaction end product such as 12 molecules of NADPH. 12 molecules of NADPH. So in this reaction to be catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. So dear students, 12 molecules of 1,3-bisphosphoglyceric acid is converted to 12 molecules of glyceraldehyde 3-phosphate. So this reaction which is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This is a reduction reaction 2. This is a reduction reaction 2. So dear students, so in this first carbonylation reaction and in this reduction reactions are we using light reaction end products such as ATP and NADPH. ATP and NADPH. So carbonylation reactions are here only one, but reduction reactions are here two. So in these two reduction reactions are utilizing light reaction end products, such as 12 molecules of ATP and 12 molecules of NADPH. Why? Because in C3 cycle first stable compound containing 12 molecules of 3-phosphoglycerate or 12 molecules of 3-phosphoglyceric acid. So hence in this time to be using 12 molecules of ATP and 12 molecules of NADPH. 12 molecules of NADPH. These are two steps in C3 cycle or Melvin-Calvin cycle. First step are here carbonylation, second step are here reduction. Second step are here reduction. Dear students, Calvin cycle or C3 cycle already completed in two phases or two steps. Now coming to third phase, regeneration. So dear students, 12 molecules of glyceraldehyde 3-phosphate to be formed glyceraldehyde 3-phosphate pool. So in this glyceraldehyde 3-phosphate pool to be separated 6 parts. Each part of 2 molecules. Totally 6 parts. So each part have 2 molecules of glyceraldehyde 3-phosphate. This is the first glyceraldehyde 3-phosphate. This is the second glyceraldehyde 3-phosphate. This is the third glyceraldehyde 3-phosphate. This is the fourth glyceraldehyde 3-phosphate. This is the fifth glyceraldehyde 3-phosphate. This is a sixth glyceraldehyde 3-phosphates. 6 parts of glyceraldehyde 3-phosphate are here. Each part have 2 molecules. So dear students, the first part of glyceraldehyde 3-phosphate is converted to DHAP, that is dihydroxyacetone phosphate, which is catalyzed by an enzyme, that is triphosphate isomerase. The first part of 2 molecules glyceraldehyde 3-phosphate is converted to 2 molecules of dihydroxyacetone phosphate, which is catalyzed by an enzyme triose phosphate isomerase. So this DHAP, so by means 1/6th triose phosphate or 2 molecules of glycerol 3 phosphate to exported out from cytoplasm into the chloroplast for synthesis of exos, then the sugar, then the sugar or one type of carbohydrate. This is a end product of Calvin cycle. Now once again, first part of 2 molecules glycerol 3 phosphate is converted to 2 molecules of dihydroxysteroid phosphate within the help of enzyme triose phosphate isomerase by means 1/6th triose phosphate or 2 molecules of glycerol 3 phosphate is exported out from cytoplasm into the chloroplast for synthesis of exos, then the sugar, that is one type of carbohydrate. So this is a end product of Calvin cycle. And remaining 5 parts of glycerol 3 phosphate molecules, that is 5/6th triose phosphate or 10 molecules of or 5 parts of glycerol 3 phosphate are used for the regeneration of ribulose 1,5 bisphosphate. 5/6th triose phosphate or 10 molecules of glycerol 3 phosphate or 5 parts of glycerol 3 phosphate molecules are again regenerated for the ribulose 1,5 bisphosphate, that is RUBP. So now second part of glycerol 3 phosphate and third part of glycerol 3 phosphate both are condensed. So both are condensed with to be formed fructose 1,6 bisphosphate. So dear students, second part, that is 2 molecules of glycerol 3 phosphate, third part, that is 2 molecules of glycerol 3 phosphate. So this is converted to dihydroxysteroid phosphate, which is catalyzed by the enzyme triose phosphate isomerase. This DHAP and 2 molecules of glycerol 3 phosphate both are condensed with to be formed 2 molecules of fructose 1,6 bisphosphate, which is catalyzed by an enzyme aldolase. Next, coming to fructose 1,6 bisphosphate undergoes dephosphorylation by means ADP is generated to be formed fructose 6 phosphate, which is catalyzed in this reaction by an enzyme fructose 1,6 bisphosphatase. Next, coming to fructose 6 phosphate and fourth part of 2 molecules glycerol 3 phosphate, both are combined within the help of enzyme transketolase, to be formed each 2 molecules of xylulose 5-phosphate and 2 molecules of enterose 4-phosphate. 2 molecules of xylulose 5-phosphate and 2 molecules of enterose 4-phosphate. Dear students, 2 molecules of xylulose 5-phosphate to be wait for some time. There is no participated for the reactions. Only participated 4 carbon compound of enterose 4-phosphate. So dear students, in this enterose 4-phosphate and fifth part of 2 molecules of glycerol 3-phosphate is converted to 2 molecules of dihydroxysteroid phosphate. This dihydroxysteroid phosphate and enterose 4-phosphate both are combined with to be form of 7 carbon compound of pseudoepilose 1,7 bisphosphate. So in this reaction, which is catalyzed by an enzyme aldolase. Aldolase. Next, coming to pseudoepilose 1,7 bisphosphate is converted to pseudoepilose 7 phosphate by means pseudoepilose 1,7 bisphosphate undergoes dephosphorylation. By means ADP is generated. So which is catalyzed by an enzyme pseudoepilose 1,7 bisphosphatase. Next, coming to pseudoepilose 7 phosphate and the last sixth part of 2 molecules of glycerol 3 phosphate, both are combined. So within the form of so each one 2 molecules of ribose 5 phosphate and 2 molecules of cellulose 5 phosphate. So in this reaction, which is catalyzed by an enzyme transketolase. So now coming to, so in this time ribose 5 phosphate and cellulose 5 phosphate to be waiting for some time, not participated for the reactions for some time. So thereafter to be participated, so 5 carbon compound of cellulose 5 phosphate. So dear students, 5 carbon compound of cellulose 5 phosphate. So within the form of 2 molecules of ribose 5 phosphate, which is catalyzed by an enzyme ribose 5 phosphate epimerase. So thereafter, and again another 5 carbon compound of cellulose 5 phosphate. So to be form of 2 molecules of ribose 5 phosphate. Within the help of enzyme saying that is ribose 5 phosphate epimerase. Next, coming to another ribose 5 phosphate is converted to 2 molecules of ribose 5 phosphate, which is catalyzed by an enzyme ribose 5 phosphate isomerase. This is a ribose 5 phosphate epimerase. Okay, now this is a ribose 5 phosphate isomerase. Don't confuse over here. So totally how many molecules are formed? 6 molecules. So where 2 molecules of cellulose 5 phosphate plus 2 molecules of cellulose 5 phosphate plus 2 molecules of ribose 5 phosphate, totally 6 molecules of ribose 5 phosphate will be formed. So this 6 molecules of ribose 5 phosphate converted to 6 molecules of ribose 1,5 bisphosphate. 6 molecules of ribose 1,5 bisphosphate. So dear students, in this reaction are again to be utilized light reaction end product, that is ATP. How many ATP molecules are utilized? That is 6 ATP molecules. Why? Because so this organic compound are having 6 molecules. So at the same time to utilize 6 molecules of ATP. So dear students, 6 molecules of ribose 5 phosphate are utilizing light reaction end product, that is 6 molecules of ADP, which is catalyzed by an enzyme ribose 5 phosphate kinase to be formed 6 molecules of ribose 1,5 bisphosphate, that is RUBP. So 5/6 triazophosphate or 10 molecules of glycerol 3 phosphate for used again regeneration of RUBP. This is a third phase or third step in regeneration. Anyhow, C3 cycle or Calvin cycle or PCR cycle or involves 3 pages or 3 steps, that is carboxylation, reduction, regeneration. In this C3 pathway, the first stable compound 3 carbon compound of 3 phosphoglyceric acid. So hence this cycle are named as C3 cycle. And those plants which undergo only in this pathway, so these plants are named as C3 plants, such as most of the dicotyledon plants. especially tomato, chili, brinjal, and bitter pepper. So these are the some examples are here. C4 plants. C4 plants. So in those of C4 plants, so are always to be formed 3 carbon compounds in their mesophyll cells of the leaves. So in this phenomenon first observed by Melvin Calvin. So hence this cycle are named as Calvin cycle. Calvin cycle. So dear students, in C3 cycle or in C3 pathway or in Calvin cycle, the primary CO2 aspect is RUBP. In C3 cycle or in C3 pathway, the first stable compound is 3-phosphoglyceric acid, 3-PGA, 3-phosphoglycerate or 3-phosphoglyceric acid. By means in this compound are containing 3 carbons. So hence that is named C3 cycle. And one CO2 is required for 3 ATP to NADPH. So hence 6 CO2 required for 18 ATP and 12 NADPH. 18 ATP, 12 NADPH. So by means, so 12 ATP plus 6 ATP, that is 18 ATP and 12 NADPH. 12 NADPH. By means, in this Kelvin cycle, how many times to be tons? 6 tons. 6 tons Kelvin cycles are required to be form of 1 mole of glucose. So dear students, 1 mole of glucose are synthesized to be using 18 ATP, 12 NADPH molecules. 12 NADPH molecules. So dear students, carboxylation reaction only used for carbon dioxide. Reduction reaction only used for light reaction end products such as ATP and NADPH. Regeneration reaction only used for light reaction end product, that is ATP. That is ATP. Dear students, carboxylation reaction only using of the atmospheric CO2. Reduction reaction only using for the light reaction end products ATP and NADPH. How many molecules of ATP and how many molecules of NADPH? So totally equal, that is 12. 12 molecules of ATP and 12 molecules of NADPH. But regeneration steps are here. Regeneration reactions are here. Only included light reaction end product only one, that is ATP. So how many molecules of ATP? That is 6 ATP molecules. 6 ATP molecules. So anyhow, only 1/6 triose phosphate or 2 molecules of glucose is always exported out from chloroplast into the cytoplasm after some time for the synthesis of exose sugar, then to converted into the carbohydrate or one type of starch. So in this Calvin cycle, by the end products are here disaccharide sucrose or starch. Disaccharide sucrose, that is glucose, fructose and starch. So this phenomenon first to be observed Melvin Kelvin, hence this cycle are named as Kelvin cycle. So this Kelvin cycle or C3 cycle or C3 pathway or PCR cycle or RPP, reductive pentose phosphate pathway, occurs in the loose of chloroplast in mesophyll cells. Loose of chloroplast in mesophyll cells. C3 cycle only present in mesophyll cells of chloroplast. Mesophyll cells of chloroplast, especially stroma. Especially stroma. Why? Because all dark reactions takes place in stroma of chloroplast. All light reactions takes place in grana of chloroplast. Don't confuse over here. So this is about the C3 cycle or Melvin Kelvin cycle or PCR cycle. Okay, now thank you. Dear students, today we are going to discuss about the C4 cycle or H-slack pathway or C4 plants. Dear students, previous classes we will discuss about C3 cycle or PCR cycle or Melvin-Calvin cycle. So dear students, C4 plants are having special type of leaf anatomy, that is Cranch anatomy. C3 plants do not have special type of leaf anatomy, that is Cranch anatomy. Only C4 leaves to be presence of special type of leaf anatomy, that is Cranch anatomy. That is Cranch anatomy. So dear students, this is a C4 plants of leaf anatomy. This is a C4 plants leaf anatomy. So in this C4 leaf shows upper epidermis and lower epidermis. Upper epidermis is above the cuticle and lower epidermis is below the cuticle, and also larger side of the stomata and also smaller side of the stomata. Upper epidermis containing cuticle and smaller side of the stomata. Lower epidermis containing cuticle and larger side of the stomata. Upper epidermis and lower epidermis between of the occupies mesophyll cells. These are the mesophyll cells. These are the mesophyll cells. These mesophyll cells do not have intercellular spaces. These mesophyll cells do not have intercellular spaces, but are having to chloroplast. These chloroplasts are in smaller size. Mesophyll cells including polycyte parenchyma and spongy parenchyma. Polycyte parenchyma and spongy parenchyma to be constituted within the mesophyll cells. These mesophyll cells containing smaller size of the chloroplast. There is no intercellular spaces. Okay, no. This is a mesophyll cell. And at the center of the leaf containing vascular bundle such as xylem and phloem. This is a xylem, this is a phloem. Xylem and phloem includes vascular bundle. In this C4 plants of leaves to be specialized leaf anatomy, that is these vascular bundles are surrounded by one ring-like bundle sheath. These vascular bundles are surrounded by one ring-like bundle sheath cells. So these are the bundle sheath cells to be appeared ring-like structures. So this special type of leaf anatomy, that is known as Cranch anatomy. This is a Cranch anatomy. Generally, every plant of root, every plant of stem, every plant of leaf only containing at the center of vascular bundles including xylem and phloem, not surrounded by another bundle sheath, especially C4 plants of leaves such as maize, sorghum, that is sugarcane, especially monocotyledon leaves, So those are the C4 plants. The best examples are here. All monocotyledon plants such as sorghum, that is maize and sugarcane. This type of leaves shown Cranch anatomy. So Cranch means breadth, that is German word, due to the arrangement of ring-like bundle sheath cells. Cranch means breadth. So this word is one of the German word due to the ring-like arrangement of bundle sheath cells. These are the ring-like arrangement of bundle sheath cells. This phenomenon first to be observed Professor V.S. Ramdas. So Professor V.S. Ramdas, SV University Botany Professor, he is a SV University Botany Professor. So he is extensive research work on C4 plants of leaves. So extensive research work on C4 plants of leaves totally to be described about Cranch anatomy, especially sorghum and sugarcane. This is a bundle sheath cell. This is a mesophyll cells. So C4 plants of leaves to be presence of C4 acids in those of mesophyll cells including bundle seed cells. But C3 cycle only takes place in mesophyll cells of chloroplast, but C4 cycle takes place in mesophyll cells of chloroplast and bundle seed cells of chloroplast. Okay now, so these are the mesophyll cells, and these mesophyll cells are have to smaller size of the chloroplast. So this is a bundle seed cells. So these bundle seed cells are have to chloroplast. What is the differences between mesophyll cells of chloroplast and bundle seed cells of chloroplast? Dear students, so these are the mesophyll cells. These mesophyll cells containing smaller size of the chloroplast. Okay now, there is no intercellular spaces, one cell to another cell. Okay now, next, so this is a bundle sheet. By means this is a vascular bundle including xylem and phloem. The xylem and phloem, that is vascular bundle, is surrounded by one ring-like arrangement of bundle sheet. So these cells are larger in size, and chloroplast is very larger than the mesophyll cells of chloroplast. Bundle seed cells of chloroplast is larger than the mesophyll cells of chloroplast. So mesophyll cells of chloroplast is smaller in size. Bundle seed cells of chloroplast are larger in size. There is no intercellular spaces, one cell to another cell, and especially impervious to gaseous exchange. So these are the character features of bundle seed cells. So this special type of leaf anatomy are clearly shown on C4 plants of leaves. So this special type of leaf anatomy are named as cranch anatomy. In this cranch anatomy, to be especially shown on sorghum, that is maize and sugarcane. First will be described by Professor V.S. Ramdas. Professor V.S. Ramdas is a SV University botany professor. To be observed one special type of leaf anatomy, that is cranch anatomy. Cranch anatomy only shown in special type of C4 leaves such as maize or sorghum and sugarcane. So dear students, so to be detailed describe about the C4 pathway. This is a mesophyll cell. Okay now, this is a bundle sheet cell. So mesophyll cell, every cell containing cell wall and plasma membrane or cell membrane. Bundle seed cells also containing cell wall and plasma membrane. So both cells are connected by one plasma desmata. Okay now, mesophyll cells and bundle seed cells are connected by plasmodesmata with the help of cytoplasm through the plasmodesmata, one cell to another cell. Cytoplasm or organic acids to be transferred one cell to another cell through plasmodesmata. Okay now, this is a mesophyll cell, this is a bundle seed cell. Mesophyll cell and bundle seed cell is connected by, both cells are connected by one plasmodesmata. Plasmodesmata is always to be help connecting of neighboring cells and the transport of cytoplasm and some of the organic compounds in one cell to another cell. Okay now, dear students, so first already the C4 plants of leaves containing one organic compound, that is phosphoenol pyruvate. Already C3 plants of leaves, chloroplast containing C3 carbon compound of that is RUBP, by means pi carbon compound in C3 plants of leaves, in their chloroplast to be containing pi carbon compound of RUBP to be aspected by CO2. This is a primary CO2 aspector to be formed 3 carbon compound of phosphoglycerate. At the same time, the C4 plants of leaves, especially mesophyll cells of chloroplast, the mesophyll cells of chloroplast, the mesophyll cells of chloroplast, the mesophyll cells of chloroplast containing 3 carbon compound of substrate, that is phosphoenol pyruvate, short form PEP. C4 plants of leucine mesophyll cells of chloroplast already will be containing 3 carbon compound of phosphoenol pyruvate, short form PEP. This phosphoenol pyruvate to be receiving of the 1 atmospheric CO2. This atmospheric CO2 directly not receiving of the phosphoenol pyruvate to be changing one carbon form, that is bicarbonate. Atmospheric CO2 to be changing into the bicarbonate form, that is SCO3 minus. So this SCO3 minus to be receiving of the phosphoenol pyruvate within the help of enzyme phosphoenol pyruvate carboxylase. That is PEPcase. This is a fixation reaction, CO2 fixation reaction within the form of 4 carbon compounds of acid, that is C4 acid, that is named OAA, oxaloacetic acid. Okay, now C4 plants of leucine mesophyll cells of chloroplast containing 3 carbon compound of phosphoenol pyruvate. So by this phosphoenol pyruvate to be accepted by bicarbonate ion, which is catalyzed by an enzyme PEPcase, to be formed 4 carbon compound of dicarbolic acid, 4 carbon compound of dicarbolic acid, that is C4 acid. The C4 acid are named as OAA, oxaloacetic acid. This is the first stable compound in C4 pathway or C4 cycle. First stable compound are here OAA, oxaloacetic acid, and the first primary CO2 aspect is PEP, phosphoenol pyruvate. In C4 cycle or C4 pathway or Arch-Slack pathway, the primary CO2 aspect is PEP. The first stable compound is OAA, oxaloacetic acid. So dear students, so C4 pathway to be presence of the C4 cycle as well as the C3 cycle. So first to be form of C4 cycle. Why? Because to be form of C4 organic acid. In this C4 pathway to be form of first 4 carbon compound, that is oxaloacetic acid. So hence this cycle are named as C4 cycle, and this phenomenon which undergoes in this pathway only some plants, those plants are named as C4 plants. So this phenomenon first will be observed MD Arch and CR Slack. So hence this cycle are named as Arch-Slack pathway. So dear students, next coming to the C4 acid, that is OAA, oxaloacetic acid, to be transported within the plasma desmata, in bundle seed cells. By means the C4 acid of oxaloacetic acid. This is a one dicarboxylic acid. The 4 carbon compounds in dicarboxylic acid of OAA, oxaloacetic acid, to be transported within the bundle seed cells. Dear students, sometimes so in C4 plants to be form of another C4 acids such as malic acid and aspartic acid. So in this oxaloacetic acid to be transported within the bundle seed cells. So this C4 acid, that is oxaloacetic acid to be lossing of one CO2 molecule, to be removal of one CO2 molecule, that is decarboxylation, to be form of 3 carbon compound of acid, that is C3 acid. So the C3 acid are named as pyruvic acid. So dear students, C4 acid containing 4 carbon compounds, but one carbon compound to be released in this bundle seed cells. So in this one carbon compound, that is CO2, to be utilized Calvin cycle, Calvin cycle finally to be form of one end product, that is glucose or starch. Glucose or starch. By means bundle cells to be presence of C3 cycle, mesophyll cells to be presence of C4 cycle. C4 cycle. Okay now. 4 carbon compounds of dicarboxylic acid, that is OAA, oxaloacetic acid. This oxaloacetic acid to be transported within the bundle cells. So in this time the C4 acid to be during the decarboxylation process by means to be losing of one carbon. So in this carbon dioxide to be utilizing in the C3 pathway or Calvin cycle process. So again to be continuous the Calvin cycle process to be bundle cells, finally to be form of one type of end product, that is a disaccharide of sucrose or starch. Sucrose or starch. Next coming to, so to be form of 3 carbon compound acid, that is pyruvic acid. That is pyruvic acid. By means bundle cells are having to C4 acid and C3 acid. That is oxaloacetic acid, either malic acid, either aspartic acid, and 3 carbon compound acid, that is pyruvic acid. Pyruvic acid. So C4 acid and C3 acid between of the to be reprocess of the Calvin cycle. Calvin cycle. By means Calvin cycle again regenerated. Calvin cycle again regenerated between of the C4 acid and C3 acid. C3 acid. So next coming to, so in this 3 carbon compound of acid is transported in mesophyll cell within the help of plasmodesmata. Within the help of plasmodesmata. By means C3 acid, that is pyruvic acid, again transported within the help of plasmodesmata. Transported within the help of plasmodesmata in mesophyll cells. In mesophyll cells. The 3 carbon compound of pyruvic acid are again utilizing the ADP molecules, light reaction end products. Light reaction end products, that is ADP molecules, are again utilizing ADP molecules to be regenerated. Pyruvic acid again regenerated within the form of same phosphoenol pyruvate. Same phosphoenol pyruvate. By means mesophyll cell first to be having phosphoenol pyruvate, and at the circular flow again to be form of the phosphoenol pyruvate. So dear students, the mesophyll cell takes place in C4 cycle, the bundle cells takes place in C3 cycle. However, in C4 pathway or Ashlag pathway presents 2 types of cycles. One is C4 cycle, another one is C3 cycle. C4 cycle takes place in mesophyll cells, C3 cycle takes place in bundle cells, especially C4 pathway. But C3 pathway only takes place in one type of cycle, that is C3 cycle, only occurs in mesophyll cells. But here in this type of leaf anatomy shown in 2 types of cells, mesophyll cells and bundle cells. So mesophyll cells containing chloroplast and bundle cells are containing chloroplast. This type of chloroplast are named as dimorphism. C4 plants of chloroplast are shown in dimorphism. Mesophyll cells containing chloroplast, bundle cells containing chloroplast, but mesophyll cells of chloroplast are having 2 grana, but bundle cells of chloroplast are not having 2 grana. That is granal and agranal. So this phenomenon are named as dimorphism. But in C3 plants do not show the dimorphism. Only one type of chloroplast are present in mesophyll cells. But C4 plants leaves to be especially shown on dimorphism. Mesophyll cells also containing chloroplast, bundle cells also containing chloroplast. So this phenomenon are named as dimorphism. And mesophyll cells and bundle cells are connected by one part, that is plasmodesmata. So dear students, anyhow C4 plants are having special type of leaf anatomy, that is cranch anatomy. This is a special type of leaf anatomy, that is cranch anatomy. Cranch means wreath. Cranch means wreath. Wreath is one of the German word due to the ring-like arrangement of bundle sheath cells. Ring-like arrangement of bundle sheath cells. So dear students, primary CO2 aspect is phosphoenol pyruvate. This phosphoenol pyruvate containing 3 carbon compound. At the C3 pathway, RABP containing 5 carbon compound. So in the C4 pathway, first stable compound OAA, oxaloacetic acid. This is containing 4 carbon compound. So dear students, one CO2 is required 5 ATP molecules and 2 NADPH molecules. So ATP molecules and NADPH molecules do not clearly shown of the C4 plants of the leaves, but C3 plants of the leaves, one CO2 to be required 3 ATP and 2 NADPH. But in C4 plants of leaves, one CO2 will be required 5 ATP and 2 NADPH. So hence 6 CO2. So hence 6 CO2 will be required 30 ATP and 12 NADPH. That is 6 5 are 30, 6 2 are 12 NADPH. To be make a 1 mole of glucose, for the synthesis of 30 ATP molecules and 12 NADPH molecules. By means in this C4 pathway is also to be 6 tons of cycles will be required. 6 tons of cycles will be required. Finally will be form of 1 mole of glucose. So this 1 mole of glucose is required for the synthesis of 30 ATP molecules and 12 NADPH molecules. Anyhow, light reaction, end products ATP and NADPH, these are replaced C3 plants of leaves and C4 plants of leaves. Finally to be make a one type of carbohydrate, that is CO2. That is CO2, atmospheric CO2. In C3 plants, in C4 plants, atmospheric CO2 is reduced to one type of carbohydrate. One type of carbohydrate to be using of the light reaction end products such as ATP and NADPH molecules. ATP and NADPH molecules. Okay, now this is about the C4 cycle, H-slack pathway or C4 plants. Okay, now dear students, previous classes we will detailed discussed about the C3 pathway or C3 cycle or Calvin cycle and C4 pathway or C4 cycle or H-slack pathway. Today we are going to discuss about the differences between C3 pathway and C4 pathway or C3 plants and C4 plants. This question is very important in IP pattern. Most of the time to be repeated in this question. 4 marks are here. This question is very important are here. Dear students, listen carefully. So the differences between C3 pathway or C3 cycle and C4 pathway or C4 cycle. First difference between are here. C3 plants occurs mostly in temperate and some tropical plants. C4 plants occurs mostly in tropical and subtropical plants. Dear students, C3 plants occurs mostly temperate and tropical regions, that is tropical C4 leaves to be a having special type of anatomy, that is known as cranch anatomy. By means vascular bundles is surrounded by one ring-like arrangement of bundle sheet, that is called cranch anatomy. So this cranch anatomy to be clearly shown of maize and sugarcane plants of leaves, to be detailed described about the Professor V.S. Ramdas. Professor V.S. Ramdas. Third difference between are here C3 plants only Kelvin cycle or C3 cycle occurs, takes place in mesophyll cells, but C4 cycle occurs in Kelvin cycle and C4 cycle. Dear students, C3 pathway only occurs in Kelvin cycle or C3 cycle, but C4 pathway occurs in two types of cells. C4 plants of leaves are containing two photosynthetic cells, but C3 plants of leaves are having only one kind of photosynthetic cell, that is mesophyll cells. Okay, now C3 plants of leaves are having only one kind of photosynthetic cell, that is mesophyll cell. So hence the Kelvin cycle is always takes place in mesophyll cells. But C4 plants of leaves containing two kinds of photosynthetic cells. One kind of photosynthetic cell, that is bundle sheath cell. Second kind of photosynthetic cell, that is mesophyll cell. Mesophyll cell. But here, so in C4 pathway, presence of two types of cycles. One is C3 cycle as well as C4 cycle. So C3 cycle occurs only bundle sheath cells, but C4 cycle occurs in mesophyll cells. Mesophyll cells. Don't confuse on here. C3 plants of leaves are having only one kind of photosynthetic cells, that is mesophyll cells. Only Kelvin cycle. But here C4 pathway. So C4 plants of leaves are containing two kinds of photosynthetic cells. One is bundle sheath cells, another one is mesophyll cells. Bundle sheath cells is also presence of C3 cycle. Mesophyll cells is also presence of C4 cycle. C4 cycle. So reversible are here. Fourth difference are here. Chloroplast is not dimorphism. Cells only have one type of chloroplast. Dear students, mesophyll cells are having only one type of chloroplast, that is grana type of chloroplast. That is grana type of chloroplast do not show two types of chloroplast, do not show dimorphism. But C4 plants of chloroplast is present dimorphism. Cells will having two types of chloroplast. One is granule and another one is agranule. C4 plants of cells are having two types of chloroplast. Any cell containing two types of chloroplast, that is called dimorphism. Any cells are containing two types of chloroplast, that is called dimorphism. So one cell is mesophyll cell, another one is bundle sheath cell. Mesophyll cells are also present chloroplast. Bundle sheath cells are also present chloroplast. But here mesophyll cells of chloroplast are smaller in size. Bundle sheath cells of chloroplast are here larger in size. Larger in size. And mesophyll cells of chloroplast containing grana, but bundle sheath cells of chloroplast are not containing grana, that is agranule. That is agranule. This is very important area. Next, fifth difference between are here. In C3 plants, the primary CO2 aspirant is RUBP. In C3 plants or in C3 pathway or in Kelvin cycle, the primary CO2 aspirant is RUBP. RUBP means ribulose 1,5-bisphosphate. Ribulose 1,5-bisphosphate presence of chloroplast containing 3 carbon compound of PEP. This PEP to be aspirated by high efficiency of atmospheric CO2. Next, eighth difference between are here. In C3 plants, photorespiration is very high. In C4 plants, photorespiration is not detectable. So in C3 plants, photorespiration is very high. Photorespiration to be combined with photosynthesis and respiration. Photosynthesis is one of the anabolism process. Respiration is one of the catabolism process. In their photosynthesis process, to be released end product of oxygen, at the same time to be released some amount of the carbohydrate. But in their respiration, to be used oxygen or carbohydrate, to be released some amount of the energy. This is a catabolism process. But here in C3 plants and C4 plants are involved photorespiration. So photorespiration means this is a one wastable photosynthesis process. This is a one photosynthesis waste process. Photorespiration means this is a one photosynthesis waste process. Generally photosynthesis to be release of O2, but in this time to be release of CO2. Okay now, photorespiration is very high in C3 plants. Photorespiration is not detectable or very low in C4 plants. Next, coming to ninth difference between are here. In C3 plants, photosynthesis yields in low to average. In C4 plants, photosynthesis yields in very high. So photosynthesis yields is always in C3 plants low to average. In C4 plants, photosynthesis yields is very high. Why? Because photorespiration is not detectable and photorespiration is very high. In this time, photosynthesis yield to be decrease. So next, 10th difference between are here. C3 plants and C4 plants. In C3 plants, the optimum temperature for this pathway is 15 degrees centigrade to 25 degrees centigrade or Celsius. In C4 plants, the optimum temperature for this pathway is 30 degrees Celsius to 45 degrees Celsius. In C3 plants, the optimum temperature is 15 degrees Celsius to 25 degrees Celsius. In C4 plants, the optimum temperature is 30 degrees Celsius to 45 degrees Celsius. This is very important difference between are here. Next, 11th difference between are here. In C3 plants, the CO2 composition point is very high. In C4 plants, the CO2 composition point is very low. CO2 composition point is very high in C3 plants. Why? Because this is easily absorbable, less efficient of atmospheric CO2. So CO2 composition point is very low. Why? Because this is absorption of atmospheric CO2 is high efficient. Very high efficient. Last and 12th difference between are here. In C3 plants, for the synthesis of one glucose molecule is required 18 ADP and 12 NADPH. 18 ADP and 12 NADPH. In C3 plants, to be make one glucose for required 18 ADP and 12 NADPH. Okay now. So in C4 plants, for the synthesis of one glucose molecule is required 30 ADP and 12 NADPH. In C4 plants, so to be synthesis of one glucose molecule for using 30 ADP and 12 NADPH molecules. These are the mainly differences between in C3 pathway or C3 cycle and C4 pathway or C4 cycle. Okay, now thank you. Dear students, today we are going to discuss about the CAM pathway or Dear students, CAM means cross-relation acid metabolism plants. Cross-relation acid metabolism plants. In CAM plants is an alternative to C3 pathway and C4 pathway for fixation of CO2 found in some plants, especially dry and hot climatic areas plants, such as some of the succulent xerophytic plants, especially Botany-Ii, and some of the Botany-Ii family members. CAM means cross-relation acid metabolism. This CAM pathway is an alternative to C4 plants and C3 plants, or C4 pathway and C3 pathway. CAM pathway is an alternative to C4 pathway and C3 pathway for fixation of CO2. Next, these CAM plants are here. All are succulent xerophytic plants, especially cocktail members, and in those of some Botany-Ii family members. Botany-Ii family members. Okay, now next coming to CAM pathway, most similar to C4 pathway. CAM pathway most similar to C4 pathway. Why? Because C4 plants first primary CO2 aspect is PEP, at the same CAM plants of first primary CO2 aspect is PEP, and in C4 plants the first stable compound is OAA. At the same the CAM plants first stable compound is OAA, that is oxaloacetic acid. So hence CAM pathway most similar to C4 pathway. By means CAM metabolism and C4 metabolism both are similar. Primary CO2 aspect is similar. First stable compound is similar. Next coming to some of the differences between C4 plants and CAM plants are here. C4 plants have two kinds of photosynthetic cells. Already previous classes we will detailed discussed about the C3 plants and C4 plants, but C4 plants of the cells particularly containing two kinds of photosynthetic cells. One is basophil cells and the second one is bundle sheath cells. Okay, now, but CAM plants have only one kind of photosynthetic cells. CAM plants have only one kind of photosynthetic cells, that is basophil cell. Same C3 plants is also have containing only one kind of photosynthetic cell, that is basophil cell. So hence CAM pathway is an alternative to C3 pathway and C4 pathway. C3 pathway and C4 pathway. Next coming to in CAM plants, in which CO2 is fixed during night time and used to make glucose during daytime. This is very important area. Generally in C3 plants, such as all dicotyledon plants, in C4 plants, such as especially some of the monocotyledon plants, in CAM plants, especially some succulent xerophytic plants. So in C3 plants and in C4 plants, all are mesophytic plants. In mesophytic plants of stomata, opening during daytime and closing during night time. But in all succulent xerophytic plants of stomata, opening during and stomata closing during daytime. During daytime. This is very very important here. Once again, all mesophytic plants such as dicotyledons and monocotyledons, the stomata opening during daytime and the stomata closing during night time. But all succulent xerophytic plants or all CM plants, the stomata opening night time, the stomata closing daytime. The stomata closing daytime. This is very very important here. In CIM plants, the CO2 fixation is during night time and used to make glucose or one type of carbohydrate. One type of carbohydrate during daytime. During daytime. This is very very important here. So, succulent xerophytic plant of stomata is reversible to mesophytic plants of stomata. Don't confuse here. All mesophytic plants, stomata opening daytime, closing night time, but all succulent xerophytic plants, stomata opening night time and closing daytime. Next, coming to in C4 plants, CO2 fixation and Kelvin cycle are separated in space. In C4 plants, the fixation of CO2 and Kelvin cycle are separated by means already the C4 plants of leaves are containing special type of cranch anatomy, at the same time containing two types of photosynthetic cells. One is mesophyll cell, second one is bundle sheath cell. So in their mesophyll cell, so to be containing 3 carbon compound of phosphoenol pyruvate, to be aspected atmospheric CO2, that is fixation of CO2. So this is a primary CO2 aspector, PEP is a primary CO2 aspector. So hence fixation of CO2 in mesophyll cells. But Kelvin cycle occurs in the bundle sheath cells. How? By means the oxaloacetic acid, 4 carbon compound of oxaloacetic acid to be lasting of 1 carbon, that is decarbonation, to be formed 3 carbon compound of pyruvic acid. So in this time to be CO2 involved in the Kelvin cycle process, finally to be resulted one type of starch material. One type of starch material. So dear students, in C4 plants, fixation of CO2 and Kelvin cycle, so both two are separated in space. By means fixation of CO2 occurs in mesophyll cells, Kelvin cycle occurs in bundle sheath cells. So that is separated in space. But while in CM plants, they are separated in time. Okay now. So mesophyll cells are having 3 carbon compound of phosphoenol pyruvate, then after converted to 4 carbon compound of oxaloacetic acid. Both are occurs in mesophyll cells, but Kelvin cycle takes place in bundle sheath cells. But in CM plants, so only separated by between of the time, that is night time and daytime. In CM plants, they are separated in time, that is night time and daytime. So dear students, night time, so to be formed 4 carbon compound of oxaloacetic acid, then will be converted 4 carbon compound of malic acid. By means night time to fixation of CO2. Night time to fixation of CO2 to be form a 4 carbon compound of malic acid. But daytime, so in this 4 carbon compound of malic acid converted to 3 carbon compound of pyruvic acid. So in this between of the time, so to be make a glucose, to be make a starch, by means Kelvin cycle. So takes place in daytime. Okay now, C4 cycle takes place in night time. By means CA plants is also containing C4 C4 cycle and C3 cycle. C4 pathway containing C4 cycle and C3 cycle, at the same CAM pathway containing C4 cycle and C3 cycle, but they are separated in time. So night time to be make a C4 cycle compounds, daytime to be make a C3 cycle organic compounds, C3 cycle organic compounds, and the end products to be released daytime. The end product CAM pathway or CAM plants of the end product to be released in daytime. Don't confuse over here. C3 pathway only occurs in C3 cycle, but C4 pathway occurs in C4 cycle and C3 cycle. C4 cycle takes place in mesophyll cell, C3 cycle takes place in bundle cell cell. But CAM pathway C4 cycle occurs in night time, C3 cycle occurs in daytime. By means the end product of CAM pathway, that is glucose or one type of starch to be released daytime. Okay now. And primary CO2 aspect is 3 carbon compound of phosphoenol pyruvate. In CAM pathway, first stable compound is 4 carbon compound of oxaloacetic acid. 4 carbon compound of oxaloacetic acid, that is OAA. Dear students, CAM pathway is an alternative to C4 pathway and C3 pathway, but here CAM pathway most probably to be similar of C4 cycle. So next coming to a diagrammatic representation of CAM pathway or CAM cycle. Dear students, so this is a stomata opening in night time and stomata closing daytime. Generally, stomatas are present in upper epidermis of leaves and lower epidermis of leaves, but more stomatas or succulent stomatas are containing lower epidermis of leaves, especially to be appeared leaves. So the monocot root of epidermis, the monocot stem of epidermis do not containing stomata, especially the monocot leaves of epidermis, the dicot leaves of epidermis are containing stomatas, but stomatas most of the present in lower epidermis of the leaves. So dear students, stomatas is always present in upper epidermis, either lower epidermis of the leaves. These stomatas is always help to gaseous exchange. So receiving of the CO2 O2 and coming out of the CO2 and O2. So dear students, stomata opening in all succulent xerophytic plants or in all CAM plants, the stomata opening night time and the stomata closing daytime. So first see here, let us know. So the atmospheric CO2 is formed to 1 by carbonate ion, that is HCO3 minus. Already the CAM plants of leaves of mesophyll cells containing 3 carbon compound of phosphoenol pyruvate. So in this phosphoenol pyruvate to be aspirated by atmospheric CO2, either bicarbonate ion. So this is a primary CO2 aspirator. So the 3 carbon compound of phosphoenol pyruvate then will be converted 4 carbon compound of oxaloacetic acid, which is catalyzed by the enzyme PEP-case or PEP carboxylase. This is the first reaction area. 3 carbon compound of phosphoenol pyruvate is converted to 4 carbon compound of oxaloacetic acid, which is catalyzed by the enzyme PEP-case or phosphoenol pyruvate carboxylase. Next coming to 4 carbon compound of oxaloacetic acid is converted to 4 carbon compound of malic acid. So in this malic acid, so to be formed in the mesophyll cells. So 4 carbon compound of oxaloacetic acid are replacing light reaction end product. That is NADPH. Which is catalyzed by an enzyme malic dehydrogenase. The mesophyll cell So waiting for some time, the basophil cell of vacuum. So and after some time, the 4-carbon compound of malic acid to be transferred at the daytime to be releasing. The vacuum to be releasing 4-carbon compound of malic acid in the daytime. So the 4-carbon compound of malic acid is converted to 3-carbon compound of pyruvic acid or pyruvate. Okay, in this time again to be utilizing light reaction end product such as NADPH, so which is catalyzed by enzyme malic enzyme or pyruvate decarboxylase. Why? Because 4-carbon compound of malic acid to be lost or to be dissolved of 1-carbon compound, that is fixation of CO2. This fixation of CO2 again to be continuous of the Calvin cycle. Through Calvin cycle to be make a 1 mole of glucose or starch. 1 mole of glucose or starch. Anyhow, one type of food material or 1 mole of glucose or starch to be made in daytime. All succulent xeropodic plants or all CAM plants. Okay now. Thank you. This is about the CAM pathway or CAM plants. Dear students, previous classes we will discuss about the C3 pathway and C4 pathway and CAM pathway, and also the differences between C3 pathway and C4 pathway. Now students, today we are going to discuss about the photorespiration or C2 cycle or glycolate cycle. Dear students, photorespiration. So photorespiration. So here two processes are involved in this photorespiration. One is photosynthesis, second one is respiration. In the photosynthesis process, generally uptake of CO2 and release of O2. By means uptake of carbon dioxide and releasing of oxygen. And in the respiration process, uptake of oxygen and releasing of carbon dioxide. This is a common phenomenon. But in the photorespiration process, the uptake of oxygen and release of CO2. So the photorespiration are also known as C2 cycle. Why? Because so two molecules of RUBP uptake of 2 molecules of oxygen, at the same time to be formed 2 molecules of phosphoglycerate and 2 molecules of phosphoglycolate. By means every organic compound to be containing 2 molecules at the same time 2 carbon compounds, that is known as C2 cycle. And a glycolate cycle. So in this photorespiration process, the first stable compound are here 2 phosphoglycerate and 2 phosphoglycolate, but 2 molecules of phosphoglycolate, once dephosphorylation to be form of 2 molecules of glycolate. This is a photorespiration common first stable compound, that is glycolate. So hence the cycle are named as glycolate cycle. Dear students, what is meaning of the photorespiration? So here the photorespiration is defined as the uptake of oxygen and releasing of carbon dioxide by plants. Uptake of oxygen and releasing of CO2 by plants through 3 cell organelles such as chloroplast, peroxisome and mitochondria. Photorespiration process only involve C3 plants, not involved in C4 plants. So why? Because in C4 plants of cells are containing higher concentration of carbon dioxide. In those are two kinds of photosynthetic cells. One is basophil cells, another one is bundle seed cells. The basophil cells are organic compound to be uptake of high efficiency of CO2. Then after to be form some amount of organic acids, and finally to be released in the bundle sheath cells. In the bundle sheath cells, between of the bundle sheath cells, between of the oxaloacetic acid and pyruvic acid. Pyruvic acid to be releasing of higher concentration of carbon dioxide. This higher concentration of carbon dioxide to be make a 1 mole of glucose through PCR cycle. Hence, so the CO2 concentration is very high in the bundle sheath cells and basophil cells. So they does not occur to the photorespiration, especially in C4 plants, but only in C3 plants. Next, coming to in C3 plants, some O2 does bind to rubisco. So some of the bindings, 2 molecules of oxygen does bind to rubisco enzyme. Does bind to rubisco enzyme or RUBP. So in C3 plants, some O2 does bind to rubisco. Hence CO2 fixation is decreased. So in the total plant kingdom, the most abundant protein, that is rubisco enzyme. The most abundant protein or enzyme in the plant kingdom, that is named rubisco. RUBISCO. Rubisco means ribulose 1,5-bisphosphate, carboxynase and oxygenase. Carboxynase and oxygenase. By means rubisco enzyme is always to be presence of dual nature. Rubisco enzyme is always to be act of dual nature by catalyzing in carbonation reactions and oxidation reactions, especially in C3 plants of synthesis cycle or Calvin cycle, to be act of carboxylation in their photorespiration process, to be act of oxidation enzyme. So rubisco enzyme is always to be act of dual nature. One is carboxylation, another one is oxidation. Carboxylation reactions mainly occurs in the Calvin cycle. Oxidation reactions mainly occurs in the photorespiration or C2 cycle. So especially in C3 plants, some O2 does bind to rubisco enzyme. In this time, CO2 fixation is always to be decreased. Next, coming to in this photorespiration process, there is no synthesis of ATP and NADPH molecule. In their photorespiration process, there is no formation of ATP and NADPH molecules. Only replacing of ATP molecules in the glycerate. But in C3 cycle and C4 cycle and CAM cycle, to be utilizing ATP molecules and NADPH molecules for the synthesis of ATP molecules and for the synthesis of NADPH molecules. But photorespiration or C2 cycle does not utilizing of the or does not synthesizing of the ATP molecules and NADPH molecules. Okay now? So in the photorespiration process, there is no synthesis of ATP and NADPH molecules. Therefore, photorespiration is a waste process. Why? Because in their photorespiration process, there is no formation of ATP molecules and NADPH molecules. Next, coming to photorespiration process, involves in trace cell organelles such as chloroplast, peroxisomes and mitochondria. Photorespiration occurs or involves in photo respiration. Next coming to the photo respiration process or C2 cycle or glycolate cycle. So photo respiration involves three cell organelles. So this is a chloroplast cell organelle, this is a peroxisome cell organelle, this is a mitochondria cell organelle. Mitochondria cell organelle. So first now coming to the photo respiration process in the chloroplast. Okay, clearly understood. So first the chloroplast are having two molecules of RUBP. So this RUBP uptake of two molecules of oxygen in the atmosphere. So dear students, generally the air level of oxygen 21%, the air level of carbon dioxide 0.03%. But in this time, to be increasing of the oxygen concentration, to be increasing of the 1%, that is high rate of oxygen. So the high rate of oxygen to be bind to the Rubisco enzyme. So in this time, two molecules of RUBP aspected by two molecules of oxygen. Okay, now the chloroplast cell organelle already containing two molecules of RUBP, that is ribulose 1,5-bisphosphate. This two molecules of RUBP uptake of two molecules of oxygen in the atmosphere, and internally the two molecules of oxygen bind to Rubisco enzyme. Why? Because Rubisco enzyme to be act of oxidase, but in this process to be act of oxygenase. Why? Because air level of the oxygen 21%, air level of the carbon dioxide 0.03%. In this time there is no increasing of CO2 concentration, only increasing of the high level of oxygen. So above the 21%. So in this time the two molecules of RUBP to be formed, two molecules of phosphoglyceric acid and two molecules of phosphoglycolate. So dear students, two molecules of phosphoglyceric acid or two molecules of phosphoglycerate again through the PCR cycle to be formed of starch, but only involved two molecules of phosphoglycolate. So the two molecules of phosphoglycolate to be dephosphorylation to be formed two molecules of glycolate. So two molecules of glycolate, so within the form of chloroplast, the chloroplast of glycolate will be transferred into the peroxisome, another cell organelle, that is peroxisome. So the two molecules of glycolate to be transferred into the peroxisome cell organelle. By means the chloroplast containing two molecules of glycolate, this two molecules of glycolate to be transmitted into the peroxisome cell organelle. So next coming to the two molecules of glycolate to be formed two molecules of glyoxolate. Why? Because so two molecules of glycolate, so within the utilizing of oxygen. Why? Because two molecules of glycolate are to be containing internally two molecules of hydrogen peroxide. This hydrogen peroxide to be break down to be release of oxygen and also water molecules. But oxygen molecules are utilized with two molecules of glycolate to be formed two molecules of glyoxolate. So which is catalyzed by an enzyme glycolate oxidase. Okay. Next coming to two molecules of glyoxolate. So to be formed two molecules of glycine. By means two molecules of glyoxolate to be formed some amino acids within the help of amino transferase enzyme to be formed two molecules of glycine. So the peroxisome containing two molecules of glycine. So by means the peroxisome of two molecules of glycolate to be formed two molecules of glycine. So one organic acid compound to be formed of one amino acid. respiration. Why? Because in their photosynthesis process, uptake of oxygen, thereafter uptake of oxygen, thereafter uptake of oxygen. Why? Because hydrogen peroxide to be breakdown or splitting into release of water and oxygen. So this oxygen also used breathing, and thereafter to be form of some amino acids. Some amino acids. So hence this is a photosynthesis cell organelle. This is a respiration cell organelle. Between of the photorespiration cell organelle. Between of the photorespiration cell organelle. Photorespiration involves three cell organelles such as chloroplast, peroxisome, mitochondria. But mainly photorespiration occurs or takes place in peroxisome. Peroxisome. This is correct answer over here. So 2 molecules of glycine again to be dissolved of carbon dioxide to be form of serine. To be form of serine. Okay, now at the same time to be released ammonia and utilizing of reduced NAD. Utilizing of reduced NAD to be form of serine. To be form of serine. So in the respiration process to be released CO2. To be released CO2 and serine. So the mitochondria to be make some amino acids such as glycine and serine. Glycine and serine. 2 molecules of glycine is converted to one type of amino acid, that is serine. So in this reaction to be releasing of CO2 to be utilizing, so glycine decarboxylase. Glycine decarboxylase and releasing of ammonia and to be utilizing of reduced NAD. So in this serine to be again transferred into the peroxisome. So this peroxisome serine is converted to hydroxyl pyruvate and hydroxyl pyruvate is converted to glycerate. Is converted to glycerate and glycerate again is converted to chloroplast cell organelle. This glycerate are again converted to phosphoglyceric acid or phosphoglycerate. Phosphoglycerate. This phosphoglycerate again through PCR cycle to be form of one type of starch molecule and to be combined 2 molecules of RuBP. And to be combined 2 molecules of RuBP. This is a common process in photorespiration. In the photorespiration mainly involves chloroplast, mitochondria, peroxisome, but mainly takes place in peroxisome. In the photorespiration process to be form a some amount of amino acids such as glycine and serine. Glycine and serine. Glycine and serine. Okay, now. So this is about the photorespiration or C2 cycle. Okay, now thank you. Dear students, today we are going to discuss about the factors affecting in photosynthesis. This is a last topic in photosynthesis. So dear students, the rate of photosynthesis is very important in determining of the yielding plants, including cropping plants. Photosynthesis is influenced by several factors, that is internal or plant factors and external factors. So first we are going to discuss about the internal factors. So dear students, internal factors include the number, size, age and orientation of leaves and basophil cells as well as chloroplast and internal concentration of CO2 and the amount of chlorophyll. The amount of chlorophyll. These are the internal factors. Especially the internal factors consist of plant body. So those internal factors include the number, size, age and orientation of leaves. Orientation of leaves. By means how many number of leaves are present, what is the size of leaves and what is the age of plant and orientation of leaves, arrangement of leaves and in which leaves containing how many types of basophil cells, and these basophil cells containing how many chloroplasts, and internal concentration of CO2, and the amount of chlorophyll. The amount of chlorophyll. These are the internal factors. And external factors. External factors include availability of light, and concentration of CO2, and temperature, and water. So these are the external factors. Mainly discussed about the external factors one by one. So anyhow, when several factors such as internal factors and external factors affected any chemical process, affected any biochemical process, so this process was to be observed Blackman's in the year 1905 to be proposed a law of limiting factors. Law of limiting factors comes into effect when several factors such as plant internal factors and external factors affected any biochemical process. This process was to be observed by Blackman in the year 1905. Then after to be described law of limiting factor. Law of limiting factor comes into effect. So the Blackman's law of limiting factors, which is the states are here. If a process that is like photosynthesis is conditioned as to its rapidity by a number of separate factors. Internal factors, either external factors, but the rate of the process is limited by the factor. The rate of the process is limited by the one slowest factor. The rate of the process is always limited by the one factor, that is slowest factor. So that is present a relative minimal value. Dear students, the rate of the photosynthesis is very important in determined by the yield plants, including cropping plants. The rate of the photosynthesis is influenced by several factors. So these several factors include internal or plant factors and external factors. Internal factors are here including the number, size, age, and orientation of leaves, basophil cells, and chloroplast, and concentration of CO2, and the amount of chlorophyll. And external factors include availability of light, concentration of CO2, and temperature, and also water. So these several factors, several factors, that is internal factors, either external factors, so affected by any biochemical process. So this process first to be observed Blackman's in the year 1905 to be described about the law of limiting factors. That is, law of limiting factors comes into effect. So this law of limiting factor, which states that the following: if a process like photosynthesis is determined to its is a rapidity by a number of separate factors, but the rate of the process is limited by only one factor, that is one slowest factor, that is present in a relative minimal value. That is the law of limiting factor. This is a law of limiting factor. This law of limiting factor is proposed by Blackman in the year 1905. That is Blackman's law of limiting factor. Blackman's law of limiting factor. Once again, what is the Blackman's law of limiting factor? So dear students, if a process by means like photosynthesis is conditioned as to its rapidity by a number of separate factors, but the rate of the process by which the rate of the photosynthesis is limited by the rate of the photosynthesis is limited by the one slowest factor, that is light. That is present is always a relative minimal value. So this is about the internal factors and external factors as well as Blackman's law of limiting factor. So dear students, next coming to briefly discuss about the external factors. External factors includes light, concentration of CO2, temperature and water. Okay. So first we will discuss about light. So dear students, in the presence of sunlight, to be utilizing carbon dioxide and water to make some amount of food material. This is a common process of photosynthesis. Photosynthesis is one type of anabolism by means smaller size of the molecules to be formed on larger size of the molecule. This is a anabolism. So in this anabolism process, to be utilizing two raw materials. First raw material are here CO2, second raw material are here water H2O, and in the presence of sunlight, to be make some amount of glucose, sucrose or one type of starch. At the same time, to be release of one end product, that is oxygen. So dear students, light is so rarely affect on the photosynthesis to be their nature. Light is rarely affect to be photosynthesis on their nature. So dear students, to be observed one graph show. So this graph showing is always effect of light intensity on the rate of photosynthesis. This is a one graph. So this graph showing is always effect of light intensity on the rate of photosynthesis. So this graph containing x-axis and y-axis, and this graph containing to be A point, B point, C point, D point and E point. So to be discussed about one by one. Dear students, first to be expressed at high intensity of light. The rate of the photosynthesis is increased by limiting some factors. The limiting factor that is light. This is a point, this is a B point. So this graph showing are here. So this graph showing are here. So A point and B point to be containing one of the curve. So this curve of A and B. So at a high intensity in the rate of the process is limited, becoming some factors, that is A region and B region. So this A region and B region limited by one of the factor, that is light. And C is always saturation of light. C point is always represent saturation of light. And D point and E point is always represent at light intensity in the presence of photosynthesis to be increasing by increasing intensity of light. That is the importance of graph. Light is rarely effect on the photosynthesis for their nature. So dear students, to be observed one light graph. So this graph showing effect of light intensity on the rate of photosynthesis. So this graph containing A, B, C, D, E points. So first to be observed at a high intensity of light. High intensity of light in the presence of photosynthesis to be affected by becomes one of the limiting factor. To be affected by one of the limiting factor. So the limiting factor are here A and B. The limiting factor are here A and B. So in this time, so the graph not showing of the linear, only showing of the to be make a one curve like structure. To make a one curve like structure, not expressed one linear structure, not expressed one linear structure to be appeared one curve like structure. Why? Because so A point and B point to be availability of limiting factor, that is light. By means at a high intensity of light in the presence of photosynthesis becomes limiting factors such as A region and B region. So the limiting factors are here light. So in this time the graph growth not to be linear. So the graph growth to be curve like structure. And C point is always represented to saturation of light, and D point and E point is always represented at light intensity in the presence of photosynthesis to be increasing by increasing intensity light. By increasing intensity light. This is a showing of one light graph. One light graph between the differences between light intensity and the rate of photosynthesis. Finally, to be applied high rate intensity but not increasing of the rate of the photosynthesis, not increasing of the concentration of CO2, not increasing of the concentration of CO2. Why? Because light is the limiting factor. Light is the limiting factor. There is no increasing of the rate of photosynthesis by means rate of the carbon dioxide. Rate of the carbon dioxide. Next, coming to concentration of CO2. Carbon dioxide is one of the major factor in the photosynthesis. Generally, the CO2 concentration is available for the atmosphere 0.03 to 0.04 percentage, but to be increasing up to 1 percentage of carbon dioxide up to 0.05 percentage, up to 0.05 percentage. So to be having to high rate of photosynthesis, high rate of photosynthesis. This consists of CO2 to be observed in C3 plants and C4 plants, C3 plants and C4 plants. But here C3 plants are having to higher concentration of CO2. At the same time, to be productive of higher, this higher productive to be using of the several greenhousing plants, such as tomatoes and bell pepper. So these greenhousing of plants grow in atmospheric CO2, and to be produced several yielding crop plants, that is the concentration of CO2. Next, coming to third additional factor, temperature. Generally, in all mesophytic plants are having to one type of temperature, that is 10 degrees centigrade to 40 degrees centigrade. This temperature is different in different organisms, especially most of the gymnosperms such as conifers. The temperature is always 6 degrees centigrade. By means coniferous is always grown only 6 degrees centigrade, but bacteria and some cyanobacteria is also grown in at 70 degrees centigrade. But C3 plants is grown at the between of the 15 to 20 degrees centigrade, 25 degrees centigrade. and C4 plants is always prone to be enzyme activity 25 degrees centigrade to 35 degrees centigrade. Okay now, so temperature is the most important part here. If the plants containing high light intensity, so hence temperature is low, but low intensity the temperature is high. This is a common phenomenon. In some plants to be containing high light intensity if the temperature is low. In some plants containing low intensity the temperature is high or optimum. So generally in all mesophytic plants are having to 10 degrees centigrade to 40 degrees centigrade, but in C3 plants are having to 15 degrees centigrade to 25 degrees centigrade. C4 plants are having to 30 degrees centigrade to 40 degrees centigrade, but conifers are grown in 6 degrees centigrade and bacteria and cyanobacteria grown in 70 degrees centigrade. Anyhow, the C4 plants easily saturation of 360 μII level, and C3 plants easily saturation about 450 μIL mm. The C4 plants easily saturation about 360 μIL power the -1, but C3 plants easily saturated in 450 μIL power -1. In C4 plants easily saturation 360 μIL power -1. C3 plants easily saturation 450 μIL -1. And water is also required for the photosynthesis process, especially photosynthesis process to be utilizing two raw materials. First raw material carbon dioxide, second raw material water. So in the presence of sunlight to be utilizing CO2 and water to be making one carbohydrate and to be releasing of water and oxygen. So this water is very essential in the growing of the plant body. Mainly, so if water stress causes closed stomata, reduced CO2 availability. If water stress causes closed stomata, in all mesophytic plants, the stomata opening daytime and the stomata closing night time. Why? Because stomata closing night time, the cause of water stress. So hence the closed stomata. So in this time, reduced CO2 availability. By means the reduced CO2 to be make one type of carbohydrate. But in all succulent deuterophytic plants, the stomata opening night time and the stomata closing daytime. Why? Because the stomata closing daytime if the causes of water stress. So hence in those plants to be making of food materials in this daytime. But all mesophytic plants the food material to be making night time. Why? Because reduced CO2 availability. So the reduced CO2 into a carbohydrate within the utilizing of light energy products such as ATP and NADPH. This is about the factors affecting in photosynthesis. Okay, now thank you.